Wait, really?
Hi, everyone. When you brush with new Tooth Tunes, it's all in your head. The toothbrush that pumps 2 minutes of your favorite music directly through your teeth, straight to your head. Featuring some of today's hottest songs. You can check them out at ToothTunes.com. When brushing sounds this good, kids keep on rocking.
Never heard of this before. Watch the DSP meme? Okay, link it. Link your DSP shit.
So, what's going on, everybody? Let's talk. I'm late today, obviously. Sadly, last night sucked. And this happens from time to time, is I'll get caught up with stuff. I'm getting caught up with stuff. And sadly, it got me kind of, how can I say? Basically, last night I had a ton of shit to do. I had to wash like 2 loads of laundry.
Hold on.
How can I say? Basically, last night I had a ton of shit to do. I had to wash like 2 loads of laundry. And obviously then I had to put the clothes away. I wasn't just going to leave them lying around the house. And then I had other shit to do. I had all these errands to do. And I didn't have time to do any of them during the daytime, so I was trying to do them at night. And one way I thought, gee, this would tire me out, right? This would get me tired so that I could go to sleep. It actually had exactly the opposite effect. I was doing all these, you know, errands and everything, and it kept making my mind think about other things to do. And it basically got the adrenaline pumping in my body. So here it was, you know, very early morning hours when I should be tired and going to sleep, and I was putting clothes away and sorting clothes out to donate and doing all this stuff. And next thing I know, I'm like, oh my god, look how late it is, and I got all caught up with all this shit, you know. It happens. And it's my, you know, obviously it is my fault. I let things get out of hand, and the thing is, you know, now I live alone, right? I don't live with anyone else who can say, hey Phil, it's freaking super late in the morning, what are you doing all this shit now, you know? And what it really is, it's kind of like what my life was like back in Connecticut before I ever live streamed. If you remember, I started live streaming in 2013. So really, I only did it for about a year or so before I moved out here to Washington and I was living with, you know, my girlfriend. So for me, it's kind of like I'm back to having my own schedule. But because I have my own schedule, I kind of just wasn't... I need to be better at keeping track of time and doing things at times when, you know, when it makes sense. And it was cool that, oh wow, I got all these chores and everything done last night. I got all this good shit done, so I don't have to worry about it tomorrow. Yeah, but then I didn't go to sleep. So here it is, like 5 in the morning, I'm in bed. I'd already been laying there for like an hour. I couldn't fall asleep at all because I had all this adrenaline still kind of coursing through my veins. And I'm like, god damn it, this sucks. So I tweeted at that point, you know, listen, I'm still awake. It's obvious I'm not going to be awake in 5 hours and then ready to stream multiplayer in particular, you know. So I ended up, you know, sleeping in about an hour, so a little bit more than usual. And I do apologize for that. I did say that the stream was starting at 11 or 11, you know, between 11 and 11:30ish, and it did not start until much later. So apologies for that. Also, I apologize because the stream itself right now started late because Twitch is having all kinds of issues today. If you're not aware, there's lots of stuff going on Twitch today, but apparently there's a ton of tournaments going on, in particular the MOBA tournaments. And everyone knows the MOBA tournaments are kind of what got Twitch its notoriety and blew it up as a video game streaming website. Therefore, that being said, you know, the website may have lots of issues today. Already, I tried to start my stream 3 times and I couldn't get it to start up. It was telling me that it was running and it wasn't doing anything. It was just fucking sitting there, you know, showing that I was broadcasting, but the stream wasn't going live. So I said, all right, I'll just keep trying. It took about 25 minutes for me to get it going, but finally I got it going, and now we're up and running, okay? So let's talk, everybody. Let's talk about what you can expect today, all right? By the way, I got a $2 anonymous tip with a troll message, which I'm obviously not going to address. And I want to say thank you to Viking, who did a 100-bit cheer. He says, don't worry, Phil, the Rain King Oku will bless the stream and everything will be fine. I don't know what that's a reference to. And then Coral Guy just resubscribed for the 3rd month in a row, so Coral Guy should have earned his silver crown. Shout out to Coral Guy and thank you. So, what are we doing today on stream, folks? We are doing Tekken 7 multiplayer. That's right. We are doing online gameplay with Paul and King, because those are the only 2 characters I've actually really focused in and played with significantly yet. And that's all we're doing today. I'm going to be trying out... I'm going to have to do all player matches because, sadly, ranked matches don't work. And I don't know, you know, when they're going to work. But I certainly wish that it would work, because I do like the ranked matches over everything, but, I guess it is what it is. We gotta play what works, right? We gotta go with the flow, and right now, it's only player matches that are working. The tournaments are also working, but the tournaments kind of suck, because you get stuck waiting for people, and, in addition, there's really no control over getting into a tournament. If you get someone with a bad connection, they can end up winning the whole fucking thing, because no one can do anything, and they just spam... you know, annoying attacks that you can't really defend or block against, so... the tournaments do kind of suck. So I'm going to be trying to do all player matches today, and we'll see how it goes. Shout out to OOOO Anonymous, who did a 100-bit cheer, and he says, hope you're feeling better. Hold on. Scrolled off. Oh, he says, hope you're feeling better, here's to a good stream. Yeah, my allergies are also bothering me, which sucks. This all started on Thursday. On Thursday, I was sleeping, and I had my bedroom window open. Now, here's what happened. The landscapers decided to come by. I guess they changed their schedule, because if you remember, they used to come by on Wednesday afternoons. You'd actually hear them outside my window sometimes on stream. So I didn't know they were coming. They came early Thursday morning when my window was open, and they cut all the grass, and all the pollen from the grass blew up into my window, right into my you know, respiratory system. And I woke up, I was like, ugh! Because immediately, you know, I have pollen allergies. And immediately, my nose congested, my throat swelled up. I was like, son of a bitch. What a pain in the ass. And since then, it's been like that. Like, my throat is still kind of messed up. Because this happened on Thursday. And, you know, it may take a while now for my system to get better. Which sucks, but, you know, I'm doing my best here. I still got the fucking nasal drip and everything. So I gotta try to get it out of my system.
Jesus.
Ugh. And it's gunky and it's nasty and I hate it. Because I got another $2 troll tip. I hope this person realizes that they're not getting their money back. If anything, I think Mort hopefully learned his lesson the other day. And hopefully this guy is, you know, realizes that if he tries to charge back any of these tips, he will not be getting his money back. I'm just saying. I got over 250 bucks from Mort. You're not going to be any different, bro. I already explained this. I warned you people. Okay. I just took a decongestant. Hopefully it dries up my sinuses here. Okay. So, I'm going to start in a couple minutes here. And it's going to be at least a 3-hour gameplay stream. I may go a little longer. It all depends on how things are going. If I'm playing and I'm having fun and I'm having good matches and everyone's enjoying the stream and, you know, it's a good session, then I may keep going. But, you know, if it's shitty and the connections are kind of lame and, you know, things aren't going so well, then maybe I won't. We'll see. We'll see how it's going out. We'll see how it's going down. We'll see how it's going down, folks. Once we jump right into it. Goddess Cleo said one of the dumbest things I've ever heard in my life, and just spammed it again in the stream chat. So I am going to address it just to let you know how dumb your question is. Goddess Cleo, who's an obvious troll, says, "But Phil, if you don't read the troll messages and shoutouts, then couldn't they say that you didn't fulfill your contract and then they could dispute it on PayPal?" No! Because there's no contract, stupid. No, stupid. No one signed a fucking contract. This doesn't exist. It would be one thing if you signed up for, like, a website and you had terms and conditions, and in the terms and conditions of the website there was a contract available that says if you send money that someone has to read a message. No! When you send me money, that's my money, period. If I read your message, that's just an added bonus of something that I do to move out of my, to go out of my way to be appreciative of the fact that someone is supporting my efforts to keep streaming or keep making videos on YouTube. There's no legal contract to do anything. If someone tips me $100 and says, "Phil is a flaming homosexual with a micro penis and a hairy beast and he smells and he's a piece of shit," and all that, I don't have to fucking read it. Are you stupid? Oh my god. Alright, so folks, that's the deal with that. I will not be acknowledging troll messages, it's that simple. Looks like we got a few other things going on. Oh, Derek, you got your crown back. What the hell? Derek, you just got your sub crown back. I don't know what happened because you didn't have it and now all of a sudden it's back randomly. What a weird glitch. You didn't have it for like a day and then it just reappeared magically.
Jesus Christ.
If you are watching my live streams, tipping me, I get that money right away, versus if you cheer with bits or you subscribe to like my Twitch channel, I don't get that for 2 months. So anyone who's doing it at this point, it's great, but I don't get that until February. It's not going to help with the tax situation. Or Teespring. Teespring I usually get right away. Within a couple days, I get the profits from that. So this, or tipping me on stream are the 3 ways you can tremendously help me get out of the situation I'm in. And I really hope if everything comes together, like I said, the streaming continues as good as it's been, and people are as generous as they've been, especially with the holidays coming up. I know some people are probably going to be more generous than usual.
Is this the reference to his hater club? Don't they call themselves like the Sons of Kojima or some shit? Or Sons of Korhal or something? No, it's like Sons of Kojima or whatever, isn't it? Oh, that was based on a picture of his. Uh-oh.
Learn some StarCraft, because it's Let's Learn StarCraft. And today's topic is going to be innovation. Take a moment to think, who is the greatest StarCraft player of all time? If you're saying Flash, then...
Wow, nice meme. Okay. Do I know how to play the piano well? If you don't know how to play the piano, then I look like I know how to play the piano very well. If you know how to play the piano, then you understand that I don't really know how to play the piano at all. That's how well I know how to play the piano. My dad apparently went in and he doesn't have any more cancer on his lymph nodes. I thought they were going to have to do radiation and shit. I thought he was fucked. But apparently they're saying that it was much better than expected. And they don't see cancer having spread to the lymph nodes. Which means I don't know if I have to go to fucking Florida now. If he's not going to die for a while longer, I can put that trip off. Thank the fucking Lord. How did I end up cleaning? Shut the fuck up. Why does this video autoplay every time I go to my goddamn channel? Gary, where the fuck is the debate video from today? God damn it. Don't let Andy cash in on all those fucking views. Andy Warski, YouTube. Fuck.
Video.
Do I want to read the comments on this? I'm a 32-year-old smack-in-the-middle centrist and a free speech absolutist. Damn. Bring me the ethno-state now. I have a clip of Tara McCarthy saying she wants to kill legal immigrants. Did I really say that? I thought I just said she wants to deport them all. Oh, and that she oh no, actually I might have said that. Because she goes as far as to say, like, we should be able to violently remove people that we consider to be here illegally, which includes naturalized citizens. So, yeah, okay. She admitted to being a feminist. Oh, no. Ethno-nationalism will happen just as easily as multiculturalism did. Multiculturalism was instituted at gunpoint. Damn. Destiny will make sure nothing gets discussed. All he wants to do is muddy water. Damn. Destiny equals cuck. Literally on the verge of losing IQ points every time Destiny opens his manlet mouth. Never subbed or donated, by the way. Damn.
14 months. Maybe at 15 you'll remember me, the feels.
Maybe. Notice how he's constantly changes the subject and moves goalposts. Seriously, how the fuck else did this turn from a discussion on race realism to a discussion on interracial dating? Does anybody remember why it turned into a discussion on interracial dating? Anybody remember the exact reason why? Who asked that question? Really makes you think, doesn't it? Huh. That's a good screenshot, actually. I like that. Wait, does anybody have the timestamp where JF asks that question? I would actually, if I could find that, I would love that. Oh, shit, hold on. Maybe we can find it? Twin study, because it's not necessarily true.
Well, for these hundreds of thousands of years.
Okay, move to, which I would agree with, versus race.
Subpopulation and like period. There's just something about it.
Okay, before we get too far down on this topic, so earlier you said if my grouping is better than random, it can give me...
To totally reject the use of race in personal choice and discrimination. I do believe that there are instances of correct personal choice that you can do based on race, which is not something that I would personally reproach. I'll give you one example, and I'd like to hear you guys on this. Is it fine for you guys to have racial preferences in your sexual preferences?
He asked it right at the start. Where is share? 15, 18.
Is it fine for you guys to have racial preferences in your sexual preferences? To have certain races of people that you prefer having sex with?
Yes, I'd say so. Because it's more of a natural.
Do not tweet out his video. Wait for your own. Oh, well, I'm making fun of a comment on his video, so I don't know. Tara McCarthy is so much smarter than Destiny. It's not even funny. Notice how Destiny didn't ask science questions when Ryan was around. Nice. Nobody believes that Destiny doesn't have a micro penis. The reality is, Destiny is completely stupid and actually thinks of himself as smart. He's literally a working example of Dunning-Kruger effect. Not just the generalized insult, but an actual example. Aw, Destiny is a total dick, and I agree with pretty much nothing he says. But to call him stupid is very inaccurate. He's unusually high-functioning for an SJW autist. There we go. Destiny is an intellectual coward. This entire debate can be summed up in one sentence. Destiny hears a compelling argument, agrees partially, moralizes about its implications of believing it completely, and then proceeds to slip back into complete denial. It's this fallacy, over and over again. Today I learned setting aside descriptive claims to argue normative claims is a fallacious appeal to consequences. Ouch. Nice job. I like how I say multiple times in the conversation that I would accept any reality presented, but that's nice. TIL, go back to Reddit. Ha ha ha. I'm on the superior platform, YouTube comments. Don't you bring up that casual shit to me. Can someone tell Destiny that hypotheticals are not arguments? His style of debating is disgusting, and a genuine debate moderator in a competition setting would shut him down really fast. He is an argumentative bully. Damn. Before Tara comes on, she's stupid. You can make her points better than her, JF. After Tara leaves, she's well spoken. Did I ever say that? If I actually said she was well spoken, then you have permission to kill me if you see me in real life. I don't think I actually said she was well spoken, did I? Wait, did JF really impregnate the retarded girl that he was dating? It's the Founding Fathers writing explicitly that only free whites should be able to be citizens of the United States. Writing explicitly. Why can't we find that anywhere in the gosh darn Constitution then? Oh, doesn't it... You think people get upset when they're like, it's right there in the fucking Constitution. It's right there in the Constitution. It says it very clearly. Like, really? Where does it say that at it? Well, if you look at the preamble, okay... Follow me here. If you look at the preamble, they use the word posterity. Now, that posterity word, if we go back into some of the earlier writings of the Founding Fathers, we might be able to put together a coherent argument that maybe posterity might be in reference to a type of white people that, right there in the Constitution, huh? You know? It's strange because when they wrote about things like, freedom of speech and what the government can and cannot do, it seemed like they were pretty clear on all those parts, but somehow they forgot to put in the part about how we should all be white and Christian, huh? That's a little bit of a Fuentes part there, the Christian nation, but darn Founding Fathers just forgot to add that important part in there. Darn. Basically, what Destiny's saying is we shouldn't recognize difference in races and treat everyone the same, even when giving them medical care, so black people don't get the care they need. I love YouTube comments. Aw, good stream. Destiny did much better than I thought. Thanks, buddy. Aw, Tara got fucked and trolled heavily when they asked her if she is American, lol. Destiny even warned everyone by saying Jeff could defend her points better than she could herself. Is this seriously the best the alt-right has to offer? Destiny doesn't even know about practically any of the science and he held his own. Ouch. Destiny is an autistic, disingenuous faggot, so it doesn't really count. It doesn't really count to say he held his own. Destiny got destroyed. All he has is his sophistry. I love how I always I always assume that when people say sophistry, they're like Stefan Molyneux fans. Do you think that's accurate? Can I see like who a person is like subscribed to? Jesus Christ. Oh, did I see Glink in there? Can you like search somebody's subscriptions? Or does it go in order? I saw Breitbart in there. Oh, we got no bullshit. Jesus. There's our boy. There's our boy in there. Tara's not very good at debating. She's like a child compared to Destiny. Damn. Leave debates to men like JF and alt hype. Ooh. This is my favorite argument from now on. Most celebrities. Imagine thinking this. This is my favorite part about Trump being elected is now when I say like, yo, look at what Leonardo DiCaprio said on global warming. They can no longer say, dude, it's a celebrity opinion. They don't count. You elected a celebrity to president, my dude. Now all celebrity opinions can be talked about. That's my favorite meme about Trump being elected, is that I can quote whatever random dumb fuck celebrity I want because they voted for one in president. Oh, it feels good. Or for president. Race is more than phenotypic. That's strange because phenotypes are everything. But, okay. Beyond that, Destiny keeps using the word phenotype and then refers to genetic traits rather than phenotypes. The expression of a genetic trait is a phenotype. No? Am I... phenotype, is it not the expression of a gene? A phenotype is the composite of an organism's observable characteristics or traits, such as its morphology, development, biochemical or physiological properties, behavior, and product of behavior. A phenotype results from the expression of an organism's genetic code. Okay. I wonder what he thinks it is. The great mysteries. Destiny was right about one thing. Terra is truly a dumb hole. Destiny's such a scummy debater. African immigrants make up a small... Wow, thanks. Here, Destiny, proven what Tara was saying, that illegal immigration from Africa is increasing. Not that it was needed, since it's obvious. African immigrant population in the U.S. steadily climbs. My favorite meme. Now, I don't want to jump the gun here, because maybe this article has some merit. But I wonder, when I read this headline, I wonder if they're equating all immigrants as the same, as in immigrants and illegal immigrants are the same. Let's find out. Let's read this, and let's see if it actually talks about illegal immigrants. Because if it doesn't, wouldn't that just be pretty spicy, that they just like conflate immigrants and illegal immigrants as all the same group? But let's find out, okay? Maybe they do talk about illegals. African immigrants make up a small share of the nation's immigrant population, but the overall numbers are growing, roughly doubling every decade since 1970, according to the new Pew Research, blah, blah, blah. There are 2.1 African American... It's home to 85. The growth is evident among recently arrived immigrants, black immigrants, transatlantic slave, African immigrant... Oh, Control F, illegal. Oh, no. Awkward. Where in that link does it talk about illegal immigration? Ooh. Oof. I don't think Tara wouldn't have tweeted that herself, right? I heard somebody talk about how she tweeted a Pew article, right? You don't think, oh, no. Oh, no. Tara, why would you do that? Oof. Ooh, that's a good shit post right there. Oh, no, she blocked me on Twitter. What a dumb cunt. Dumb asshole. I feel like I don't call enough guys cunts, so I have to like balance it out. Remind me to call a guy cunt the next time I love that she blocked me. Hold on one second. She blocked everyone who tweeted at her. Sorry, hold on. Why did Tara block me? Probably because she's scared. Because she knows that I will fuck her in the ways that only my superior, throbbing Cuban dick can. That's why. Okay. Oh yeah, were there any more interviews being scheduled, Mike Spam? Do we think Mueller could still get fired, or do we think it's probably not happening? All right. It looks like even potential Texas House of Representatives. Wait, what? Jesus. All right, let's keep going. Is Destiny capable of talking like an actual human being? Descriptive claim? Normative claim? Real people don't talk like that. It's like listening to a Martian. They're useful terms for debate. Sorry you can't keep up, dude. I can't understand their meaning. I think he meant to say I can understand their meaning. It just sounds pretentious as fuck. As I said above, I know the meaning. Oh, he meant to say I can understand their meaning. But to use those terms just comes across as pretentious. He doesn't actually understand the terms he uses, but he's just using them to obfuscate the discussion and mask his shit argument behind sounding intelligent. There's a reason why genuinely intelligent people like JF and Ryan don't use terminology in every other word. And no, the way Destiny uses them is definitely excessive. Using them to base your arguments or to enhance your arguments is fine, but when you're using them in every fucking sentence, let alone in incorrect ways, the way he uses biology terms at times is pretentious.
Amazing memes tonight. Best meme of the night is when Tara immediately fell back onto the bell curve the moment you found a flaw in her argument.
Come in on scholarships and things like that. It's like they are the elite of the elite.
No, that's exactly what I was just saying.
Hey, Tara, may I ask a question?
Wait, because I want to finish this. Because there's a big problem. She acknowledges that there's a bias in the selection of the group coming to the United States because these are the best and brightest, right? These are the best and brightest. So, of course, Stephen, those people are going to be doing better. But then when you look at Europe and you go, well, look at all of these illegal immigrants, do you think middle-class people from sub-Saharan Africa or upper-class people are the ones illegally immigrating? No, of course not. You're applying a similar selection there where you're selecting for the lowest and most desperate people there as well. Why don't you acknowledge that?
If you take an average sample of sub-Saharan Africans and
The bell curve. Fucking bitch. Don't answer the question. Get fucking dumpstered. Tara sucks at debating. I'm glad we got Tara to look bad. One more. One more notch, boys. Okay. Are we ready to play Factorio and listen to our memes? Uh-oh. Why did Japanese feudalism fail? They weren't multicultural. Neither was Sparta, and it fell. Also, what about Persia? Uh-oh. This had 27 responses. Okay, I don't want to read this meme history shit. Race realism is correct in certain aspects. For example, sickle cell anemia. I keep saying anemia. Fuck. For example, sickle cell anemia is an issue only found with sub-Saharan Africans. But then he says, AKA black people. Does he think that black people only come from sub-Saharan Africa? That there are no other types of black people in any of the other countries in Africa? Or even like people in the Middle East, some people would consider like really dark. I don't know. Destiny's a piece of shit. If the racism of today is so low-key and subtle, why is it even a problem? I love white people, dude. I love white people. Okay, can we play Factorio now? I'm good with these. I use meme for every other word, Bad Bunny. I'm sorry, I can't help it. I probably overuse meme. It's gonna get me in trouble one day. Do you know why I pulled you over, kid? I don't know, dude. I was memeing pretty fast on the interstate, officer. I'm sorry. I haven't watched the stream yet, but considering JF is an actual fucking scientist and Destiny is a neat playing video games for a living, I'm going to assume that Destiny is going to need a lot of lube for his anus of this. Oof, 37 minutes ago. That guy's going to be pretty disappointed. All right, are we good on these? Holy shit, I thought this was just a 10-minute video. This is a fucking 12-parter? Holy fuck, I didn't know that. Alright, well, this will be after our intro course, I guess. What are we on, part 3 now? These better be picking up. No, we listened to part 3. We're on part 4 now, right? behavioral evolution 2.
Would be an example of this. Here you have a species in which males are migratory. It's a tournament species. A male shows up during the mating season.
We're on molecular genetics. Whoa! Well, oh, we're not listening to this in 2 times. I only did that last night because I had to sleep pretty soon. But we can take a little bit longer tonight. I'll be right back. I'm going to get a bowl of cereal. I've earned one, okay? I like cereal. I'll be right back.
Destiny is gone. He can't ban me now. They are blood type aliens. 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 ECHECHECHECHE. In 5, 4, 3, 2, 1. Ye boy.
My dudes.
Stanford University.
So we pick up from whatever the last day was, and we do our first disciplinary leap as promised at the very beginning of the course. The entire function of the first half of the course is to leap from one bucket to another, such that just as soon as you feel like you are getting mildly comfortable in one, we will spend time trashing it from the standpoint of a different discipline. This is our first one of these. And the whole point of this jump is to show on a certain level exactly where one discipline's notion of an explanation ends, another one begins. And what this one will be about is how evolution works down on a molecular level. Where did we leave off after finishing our overview of the evolution of behavior? We finished with a bunch of criticisms at the end, criticizing sort of what some of the basic tenets were of that view. First off, that notion of heritability. Heritability, assuming that all sorts of behaviors have a genetic component, have a genetic basis, have a genetic cause, and we will see those are worlds of differences in those words there. And what we saw was that that could be the case, that could not be the case. That's going to be what we focus on a great deal today. Another one of the basic tenets, this notion of adaptation. Everything you see is wonderfully adaptive. The outcome is exactly the process that evolution brings about to optimize results. The counter view being the world of spandrels. A lot of the time, stuff gets carried along as baggage. Things evolving aren't necessarily things that have been sculpted into being as adaptive as possible. Another critique, and one that's also central today, is that whole emphasis on the gradualism, on small incremental changes. And what I alluded to at the end the other day is going to see a viewpoint where something very, very different is proposed to be going on, one that will make sense only eventually when we see some pretty unexpected things about genetics and the molecular biology of. Okay, so starting off, what we also finished with was a notion that there are political agendas that run through every aspect of this subject. And if that very first lecture back when, talking about how some of these viewpoints influence who gets a lobotomy, who winds up exterminated, who winds up being viewed as educatable or uneducatable, etc. Political views run throughout all of this. You will see very strongly in this realm. Okay, so that viewpoint of the sociobiologist, evolutionary psychologist the other day, one of the tenets going after the notion of heritability. The notion that these behaviors are heritable, are genetically influenced, blah, blah. How would folks in that business do it? What they would do is exactly what we were seeing, which is say, okay, here's some behavioral structure, and we know how evolution works, and evolution of behavior, and individual selection, and kin selection, and reciprocal altruism, and evolution of, and all the above. And then they say, well, with that framework, that explains the behavior pretty well. Go show me something better. Show me a better theoretical structure that's even more explanatory, more predictive, and until you come up with something better, I'm going to assume that this one is right, and this one comes with a large assumption of heritability of genetics. All built around inferentially. These are highly structured models built around how genes work. They explain what we see with these behaviors, and until you can give me a model that does even a better job, I'm sticking with this, and this counts as my evidence for a genetic component to this set of behaviors. And what we'll see is that's exactly where the molecular biologists decide that they finally have gotten free of people doing, I don't know, poetry for all the science in it. My God, that counts as science, coming up with a bunch of these rules there and say, until you come up with fancier rules or a more pleasing just so story, I win. That counts as science. Complete contempt for this approach. This is where a more molecular stance about it all takes off. And what we'll see that not only will have lots to say about adaptiveness, it will also have lots to say about gradualism. Okay, so starting with it, in order to make sense of this, for a molecular biologist, what is evolution about? When you see traits that have evolved, what's it about? By necessity, well, one is immediately talking about our genes. genes, in this case, not as some constructs that one wants to maximize copies of and ones that your cousins have only whatever percentage of in common with you, but genes instead molecules, genes as information, genes as strings of DNA. And I'll assume by now there's like a basic level of shared knowledge about this after hearing that a lot of folks came to the catch-up session last week, which I think is a good idea. But
good job, guys.
All built about
give yourselves a pat on the back for going to the catch-up sessions. Everybody give yourselves good job. You're not just some loser freshmen, sophomores trying to knock out your gen eds. You went to the catch-up classes because you really wanted to learn this multidisciplinary approach to human behavioral biology. Give yourselves a pat on the back. Good job, guys.
All of this notion of genes eventually way out the other end having something to do with behavior is the intermediary of proteins. Proteins are important. Proteins are important not just to have in your diet and make you run fast and strong and all. Proteins are in lots of ways the structurally most important things you've got making up cells. Proteins have endless roles. Proteins hold the shapes of cells together. Proteins form messengers, hormones, neurotransmitters, all of this to come. Proteins are the enzymes that do all sorts of the most important stuff. Proteins, et cetera, et cetera, et cetera. Proteins are the workhorses of what have cells doing what they're supposed to do. So the question, of course, becomes what codes for proteins? And this is where genes come in. Our basic issue here is this flow of information. Genes specify proteins. Proteins are made up of constituent building blocks, amino acids. There's approximately 20 different ones that are commonly occurring, and each one has to be coded for with a different DNA sequence. A different DNA sequence of 3 letters, 3 nucleotides. And I hope I'm not hitting a level here where people for whom this is new, this is going way too fast, and everybody else, this is way too boring, but hang on for a while in that case. Okay, so DNA codes for amino acids. A long string of DNA coding for a sequence of them will code for a sequence of amino acids which get plugged together, and you then have a protein. You got one intervening step, which for our purposes is like not really interesting and we could ignore for the most part, which is there's an intermediary step. Genes at the level of DNA, sequences of DNA, first specify an intermediate form called RNA, and it's from that you get the readout forming the proteins. Now, everything about the function of this is built out of the following sequence. If you know the sequence of DNA, you will know the sequence of RNA. You will know the amino acid sequence. You will know the protein that thus is made.
You can probably safely 2 times this one. He just goes over parts of genes. I could probably use this brush up. Yeah.
You will know the shape of the protein, and you will then know the function of the protein. And that is this critical link between what DNA, genes, evolution, blah, blah, are about, and the actual things that pop out into the other end and do something. For the very critical reason that everything about protein function is built around shape. Here is a cliche that is required by law to be said at this point, which is all sorts of effector proteins fit into other molecules, other effector proteins like okay, everybody say it out loud. Everybody know this cliche? It fits in like yes, okay, cliched education at its best, like a lock and key. For those of you who are not tortured with this one from early on, it's the whole notion that a shape of a protein imparts information insofar as it interacts with something else of a shape which modifies it, which complements it, whatever. The whole world that we will have of various hormones and neurotransmitters will consist of hormones and neurotransmitters going into receptors where there's a critical relationship in the shape between the messenger and the receptor, and all of this is driven by proteins, protein shape, etc. All of this is driven thus by DNA sequences. Shape is everything with this. What you get from that is, of course, the question of where do you get different shapes from? And those 20 amino acids, for our purposes, for the purposes especially people without a strong chemistry background, all that's pertinent here is the 20 different amino acids have different degrees of being attracted to or repelled by water, which most proteins are spending their life swimming around in water. They are pulled towards water, they avoid it, they are hydrophilic, hydrophobic. All that means is different amino acids gets pulled into different positions by their relationship with water, and thus a string of amino acids, the shape it winds up getting with is determined by that amino acid sequence. There's a whole world of really interesting, horrible neurological diseases called prion diseases, which show that everything I just said is wrong, but for our purposes, everything I just said is right. So this is where you get this critical relationship: know the DNA sequence, know the coding, and out the other end, you will get protein shape, because of this business of different amino acids having different relationships with water, and thus strings of them coming up with different three-dimensional structures. And out of that will become coming function out of the famed lock and key nature between shapes of functions and shapes of other things. Okay, just once again for people fairly new at this, no diagram there. One of the most interesting, or the blank slate. One of the most interesting things that proteins do is when they are enzymes. Enzymes, everybody knows that enzymes are important because they put them in your laundry detergent and advertise about how cool it is that you have enzymes in your detergent. But what enzymes do is they catalyze reactions. They cause reactions to occur, which left on their own would be very, very rare events. What enzymes do is accelerate vastly, a gazillion times over, the speed with which these happen. Catalyze reactions, what do I mean by that? For our purposes, they can take two things that aren't connected and stick them together, or they can take one thing and break it apart. For our purposes, that's what enzymes do. Virtually every enzyme out there is a protein. So proteins fit into other things and send on messages. Proteins are enzymes, pull things together, pull them apart. Proteins are structural. Again, a whole lot of the superstructure of your cells are held together by proteins. This is the realm of what they do. Notice here, suddenly there's a change of shape in a protein if it's an enzyme, because what's it doing? In some way, it's got to be pulling apart something or putting something together. The shape in some cases also can change as a function of what this protein is doing with its job. We will see a classic version of that are channels. Channels in which chemicals can flow in or out of cells, ions for chemistry types, channels that will open up under some circumstances, close under others. So protein structure not only gives shape and function, but it gives the circumstances where the shape might change in a functionally relevant way. Okay, so out of all of this comes the central dogma of life, and this was proposed by Francis Crick of Watson and Crick fame. And Francis Crick was the one who formalized saying the central dogma of how life and information flows is DNA to RNA to protein. And that became defining. An entire generation of babies were told that at birth. This is the flow of information. It has been violated in all sorts of interesting ways, which will dominate a lot of what comes. But this was the central dogma. One way in which it's violated, and again, this notion is not DNA to RNA, but the notion of whatever your DNA sequence is, whatever the gene is, whatever structure of a protein it codes for, the flow of information is going to be from DNA, RNA to protein, DNA as running everything. Note the importance of that and the very statement of this as the central dogma, the canonical flow of information in life. It all starts with DNA. DNA is the one sitting here deciding when information is going to flow from DNA to RNA to DNA as knowing what's happening. And a lot of what we will see shortly is DNA knows squat. DNA is not making a whole lot of decisions there. But one simplistic way in which central dogma went down the tubes in the 1970s or so, and one that's tangential here, but just as a first blow against central dogma, one of the things that's interesting is there are things called viruses, and what viruses do for a living is get into organisms. Viruses, in the classical form, being little smidgens of foreign DNA, which are able to get into the DNA of your own, hijack the processes there, and make the cell function for its own parasitic, vicious needs. And so you're changing the starting step of how the central dogma of life, and thus you're going to change RNA and protein, all of that. In the 70s, it became apparent there was one weirdo viral world of viruses made out of RNA. This intermediate form. All sorts of people were extremely upset about this and tried to ostracize the scientists who came up with this and told them there's no way. But eventually what was shown in Nobel Prize winning glory was there's a class of enzymes. Enzymes, they pop up, a class of enzymes that could take the RNA information and turn it back into DNA viral information, and then it does its thing. Huge blow to the central, here's information running from an RNA virus, somehow being reversed back to a DNA form, and thus these are called retroviruses. Insert in the DNA, and off they go from there. So this was a major blow.
Wait, a retrovirus is a virus that starts off in RNA form and then inserts itself into DNA, and that's why it's called a retrovirus? Hold on, I have to listen to that again, I'm sorry.
Of how the central dogma of life, and thus you're going to change RNA and protein, all of that. In the 70s, it became apparent there was one weirdo viral world of viruses made out of RNA. This intermediate form, all sorts of people were extremely upset about this and tried to ostracize the scientists who came up with this and told them there's no way. But eventually what was shown in Nobel Prize winning glory was there's a class of enzymes. Enzymes, they pop up, a class of enzymes that could take the RNA information and turn it back into DNA viral information, and then it does its thing. Huge blow to the central, here's information running from an RNA virus, somehow being reversed back to a DNA form, and thus these are called retroviruses. Insert in the DNA, and off they go from there. So this was a major blow. Everybody eventually came to terms with this, but it still is a minor footnote in this Crickean world of everything flows from DNA. It is the Bible, it is the lawgiver, it is the Holy Grail, it is where it all starts. So stay tuned. Okay, so given that framework, one should immediately get mighty impressed with what if something changes in the DNA? What if one of the bits of coding is coded incorrectly? What if we now have on our hands a mutation? And what we will focus on here is classical realms of mutation in genetics
versus
and how
quantum
that plays
mutation
out in classical gradualist models of evolutionary change, and then see how all of that falls apart when you look at what's really going on. So starting off,
when he's saying classical models, is he talking about like theoretical versus like experimental or applied? What does he mean when he says classical? What is the contrast that he's pointing to? Or classical, as in like a framework within molecular genetics versus multidisciplinary, or what? Classical just means early models, old standards. Schrodinger's DNA. Classical models mean where DNA is the primary mover in all cases, but the newer theories say other things. Okay.
You can have, and again, this is going to be a review for people with a background in this, newcomers to this, hopefully this won't be going too fast, but broadly, when you were talking about this world of micromutations, for our purposes, what we will mean by a micromutation is when one letter in your DNA sequence of information gets accidentally miscopied, or gets changed by radiation, or gets changed by some chemical compound in the environment, some such thing, where there's a mistake and one letter in the DNA sequence comes up wrong. As I noted before, pairs of triplets, there we've got 3 pairs of triplets, And what we've got here is at the DNA level, a amino acid is coded for by 3 base pairs. A triplet, 3 of these, next amino acid coded for, next one coded for. So what we've just raised is the possibility of a mutation occurring. Something changing in one single one, and now asking what are the consequences going to be in classical realms of genetics and evolutionary change. Okay, broadly you can get 3 different versions of stuff going wrong. One is where one of these letters, nucleotides, translating, not essential to have that down, where one of
When he says a nucleotide, what's the difference between a nucleotide and an amino acid? How did nucleotides relate to base pairs? Amino acids are chains of nucleotides. Wait, amino acids are totally different. A nucleotide is in DNA, ACTG. Amino acids are the pieces of proteins. Nucleotides. 2 nucleotides on opposite strands make up base pairs. Okay. So ATCG are 2 nucleotides on opposite strands make up base pairs. Wait, so what is the difference between a nucleotide and an amino acid? Amino acids are pieces. Or no, wait. I'm sorry. A nucleotide is the ACTG. That's a nucleotide, right? Those 4 things are the 4 nucleotides. Wait, hold on. I just have to look this up. Let me make sure I'm getting all this terminology correct. Nucleotide is an organic molecule that serves at the Okay, A, T, C, and G are the 4 nucleotides. Gotcha. Okay.
these letters is accidentally changed into another letter, a point mutation. In some cases, point mutations are of no consequence at all. How can that be? For this very simple reason, going through some math here, there are 4 different potential letters. At each one of these sites, DNA comes in 4 different letter types, letter flavors, 4 different types of bases, and thus you can have 4 different ones in the first position, times 4, a total of 64 possible 3-step combinations of DNA letters. A possibility of 64 of them, and you're only coding for 20 amino acids. So you use up some of the 64 for signals saying stop or start or thank God it's Friday or who knows what. But what you have, though, is a large redundancy. You have a number of different triplet sequences coding for the same amino acid. There's redundancy in the genetic code. And in general, where you get that redundancy, where you get the differences in 3 triplets, a couple of different triplets that code for the same amino acid, they'll tend to differ in the middle one of the 3 letters. That's the easiest one to have a change, where it doesn't change the overall consequence of it. So you will tend to have redundancy. Each amino acid is coded for by a number of closely related triplets.
Okay. Nucleotides are the ATCG thing that make up 2 of them together are base pairs. They are bonded by some bullshit-ass hydrogen bridge. These things, these nucleotides or these genes, something of these will code for amino acids, and the amino acids are the things that are what proteins are built out of. What specifically codes for, so he's saying like 3 nucleotides will code for an amino acid that will create a protein, correct? Nucleotides and amino acids are 2 different things. Nucleotides are the building blocks for DNA, while amino acids are the building blocks for proteins. 3 nucleotides are called a codon. Codon? A sequence of 3 nucleotides in a row code. Okay, a nucleotide is 2. Wait, a sequence of 3 nucleotides. So does that includes the other things that they're paired with? Wait, nucleotides are always paired together, aren't they? Like if you have A on one side, you always have T on the other. If you've got C on one side, you've always got G on the other. That's correct, right? Or is that not true? A codon? Codon? In double-stranded DNA, yes. Codon. As long as it's DNA, yes. You only read from one strand of the DNA, so it's just the 3 in a row. So 3 nucleotides in a row are is a codon, and that codes for an amino acid, which can be used to create a protein. Okay. DNA is double-stranded, RNA is not. That's why the RNA breaks off and then can replicate. But because the nucleotides are always matched to the same nucleotide, RNA can be used to reconstruct DNA or whatever, right? Okay.
So, first possible type of mutation, this point mutation, where one letter is flipped to another. Potentially, this could be of no consequence whatsoever. If you have flipped to a letter out of the middle one, which happens to leave you with a triplet that codes for the same amino acid. In that case, you have a neutral mutation of 0 consequence whatsoever. So, that's not a very exciting mutation. In some cases, you can have a single point being changed where you wind up with a different amino acid coded for. And that's most typically if it's a mutation in here or here, rather than the boring middle one, you get a different amino acid. Is that the end of the world? Often not at all, or only minimally, because a lot of the amino acids have similar feelings and ambivalencies about water, similar responses and similar shapes. It won't be exactly the same shape, but the protein will function somewhat the same. So you could have, in this case, a point mutation, and most people would not find the second line to be impossible to make sense of in the context of the first one. Or you can have a point mutation, which by changing one letter produces a different amino acid, and this one is majorly different. This one has a completely different set of attractions or being repelled by what produces a protein of a very different shape and potentially a very different message. And shown here, just changing one letter in this case, one single point mutation, and you dramatically change the meaning of that. And I actually did that once in a grant proposal, sort of the final paragraph. We have now accomplished X, Y, and Z with your money over the last 5 years, and no wonder it didn't get renewed. So in that case, you see a single point mutation of great consequence. So you can have one of these letters, one of these nucleotides, mistaken for another, entirely neutral, moderate consequence, or major disastrous consequence. Second way of classical mutations, the second version that it could come with, is there's a deletion. One of the letters gets...
Okay, the first way was that one of the letters could be incorrect, right? That was the first letter of deletion. Or the first problem was that one of those things could be incorrect. Why am I getting attacked? Oh, fuck. My background in this more specific area is, like, non-existent. Fuck. I have to, like, actually sit here and listen to this.
Lost in the process. And what you then get is a frame shift over to...
Do I have a game that I can, like, AFK play while listening to this so we're not just staring at the lecture?
To the missing spot there, and the D from here now finishes up this. And what you can see is it suddenly becomes dramatic gibberish, and it would continue that way. A deletion mutation in classical genetics is major league. It totally changes the coding downstream from that. Third classical type, an insertion mutation, one where you now accidentally double the letter and you frame shift it in the opposite direction. Just as screwing up of meaning, major consequences there. So you have point mutations, which can have major consequences or can have none whatsoever and anything in between. You have point mutations, and then you have insertion and deletion. The last two tend to have big consequences. So all of this is built around one single base pair changing. And all of this is built around, thus, one single protein changing its shape. And thus, this world of micro mutations, single spots that are mutating, what this tends to do, and this is an important concept for what's to come, what it does is it changes how well the protein does its job. It changes the efficacy of that protein by changing the shape a little bit, by changing it dramatically, all of that. And we can see, back to our lock and key, where if thanks to a mutation, this has a slightly different shape, it will fit into the lock slightly less effectively, may stay in there for a shorter time before floating off and thus send less of a message. On the other hand, if you've got a deletion insertion that dramatically changes the shape of this, you will change how well this protein does its job. It won't do its job at all because it's going to wind up with a completely different shape and not fit in there whatsoever. What we have here is a world of mutations which are changing the function of one protein at a time. That is microevolutionary change.
Just fit into the slot, bro. LOL.
And that's what we're now going to see played out. When can this make a difference? This can make huge differences.
From DNA to protein. I just want to watch this real quick, because I didn't go to the catch-up lecture.
Here is a cell.
Yeah.
The basic unit of all living tissue.
Cool.
In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes.
Cool.
Each chromosome contains a long strand of DNA, tightly
Cool.
packaged around proteins called histones.
Histones. Okay. Hopefully we don't need to know that.
Within the DNA are sections called genes.
Cool.
These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
Cool.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed.
Duh.
This involves removing and adding sections of RNA.
Why?
The messenger RNA then moves out of the nucleus into the cytoplasm.
Okay.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding
Those 3 bases are called codons, right?
amino acid.
Wow!
This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay. So we start off with the meme. So basically, you have your DNA, the RNA comes in, the RNA reads the DNA. Once the RNA reads the RNA goes outside of the nucleus, and then a ribosome comes along, fills up the RNA to get the code, and then in the ribosome, the amino acids are created, and then once those strings of amino acids are spit out, they fold into proteins. Is that correct? Do we have it? Okay.
When it's in the realm where, thanks to a change in the shape, the protein is completely out of business. 2 examples. One where you take out the function of a pre-existing gene. First one, there is this amino acid. For our purposes, that's going to derail us. For our purposes, there's a chemical that occurs in the body called phenylalanine, which has its uses. But you don't want the levels of it to build up too high because it can become toxic, damaging to brain cells, to neurons. And fortunately, there is an enzyme made of protein which turns phenylalanine into something safer. That's all we need to know about it. So you have a mutation, in the gene coding for that enzyme.
Uh-oh.
A fancy mutation, just something in one of these categories, a classical single spot point mutation, and what you got there is an enzyme that no longer does its job. And as a result, this phenylalanine is not converted into the safer form, builds up and lays waste to one's nervous system. And thus you have a disease, PKU, phenylketonuria. Very, very common genetic disorder, and this is one where the outcome is a small mutation that has completely knocked this enzyme out of business, this enzyme which was able to turn the phenylalanine into something safer. This is not a subtle outcome. Have untreated PKU, and your reproductive success by the rules of evolution is going to be real down near zero. This destroys the nervous system very rapidly after birth. So that's dramatic. Here's another dramatic one. And this one, anyone who took BioCore, I always bring this one in as a big hormone crowd pleaser. But here's an interesting thing that can go wrong with any child you might eventually have. Okay, you've got a daughter, and she's doing just fine, and she's growing up just fine, and things are terrific, and around age 10 or 11 or so, some of her classmates are beginning to reach puberty. That's on the early side for the Western average, but it's not outrageously early, not a big deal. By about 12, statistically about half of the girls in her class have reached puberty. 12 is about the Westernized average these days. She hasn't yet, not a big deal. A year later, she still hasn't. Well, this is nothing critical, but it's getting a little bit on the late side. A year after that, 2 years after that, she has still not reached puberty. So at that point, you take her to a physician who examines her closely and figures out what's up. And at some point, the physician is probably going to have pupils dilate or some sort of weird autonomic response when they figure out what's up, and then very calmly, in a premeditated way, sit you down for a little talk afterward and inform you that your daughter has not started menstruating, has not reached puberty yet. Your daughter has not started doing this because you don't have a daughter, you got a son. This kid here, for these last 14 years, has actually been male, not female. You have what is called, no, I'm not going to tell you what, okay, it's called, especially since it's in the handout already. Anyone want to guess what it's called? It's called a TFM, testicular feminization syndrome. And you wind up with a testicular feminized male. These are individuals who genetically are male. At the level of chromosomes, that XX and XY business. These are individuals who have testes way up in their stomach or whatever, never descended down. These are people whose testes make testosterone. Testosterone out the wazoo, tons of testosterone, enough testosterone to put like antlers on your testes or something, that much testosterone, and nonetheless, you're getting a female phenotype. You are getting a female external genitalia. You are getting female everything. Yep, question? No, that's the fancy term for it. And you've just given away the punchline, you creep. Okay.
I am so glad I'm not in school anymore. The teacher's like, and anyway, sometimes, people will demand, a good a lot. Sometimes, if the cost for that good increases, people will actually still demand. Teacher! Yeah, what's up? Is this elasticity? Yeah, that's what we were just about to get to. Yeah. Sorry.
So they figured out in this disease.
What did she say? It was like androgen insensitivity or something, I think. I don't know. He'll probably say it.
There's an insensitivity to something. What could that be? Okay, go and say it again. Yes, okay. What you've got here is one of those simple little classical mutations, and what it does is it changes the shape of the androgen receptor, the testosterone receptor, and at that point it doesn't matter how much testosterone is floating around, those target cells are not going to listen. This is consequential. This is a major consequence. This is one of changing gender phenotype, and there is a long and at times absolutely appalling history of what has been done with individuals who have testicular feminization syndrome, what counts as the medically appropriate intervention. There's some horrifying history there that could be straight out of the first lecture in terms of some notions of what counts as normal gender behavior. Another version of this one's a little bit more subtle because in this case it's not wiping out the function of an enzyme, it's just making it a little bit less effective at what it does. And this has to do with a disease that is found in two different populations, slight variants on it. One is...
Do they just not go through puberty? So from the sounds of it, if you have fucked up androgen receptors, I guess what happens is you're born as a male, more or less you are a male, but the problem is your body doesn't do the things that it should in response to you having testosterone because your body's not capable of interacting with it. So even though you're producing testosterone, you don't have any of the traits of being a man, but you're never going to go through puberty as a woman because you're not creating enough estrogen. I'm not sure what signals female puberty, but yeah, so you would I assume that a person like that would just never go through puberty because they're not receptive to the testosterone their body's creating, and they're never going to go through female puberty. I don't think that would be my guess. I don't know. You go Wikipedia it. I don't actually even know why you're asking me this. Go Wikipedia it. Fuck.
Up in the Dominican Republic in the mountains. The other is in the mountains of New Guinea. Interesting similarity there. In both cases, some fairly isolated inbred populations. So that sort of has genetics written all over it. But in these cases, there's a problem with enzymes that make testosterone. So this is a whole other world. Instead of the enzyme, instead of the proteins, and thus the gene somewhere back there, that code for the receptor for testosterone, the receptor that responds to this messenger. Here, instead, it's back way up there in the testes, a bunch of these enzymes which go through a bunch of steps and make testosterone. Biosynthetic enzymes there, proteins, genes, once again. So you've got a mutation in one of those critical biosynthetic enzymes, and it's not a huge one. The shape is a little bit off. Its efficacy, the effectiveness with which it makes testosterone is down a bit, and actually it's down a lot. So here's what you wind up getting. You get an individual who, like before puberty, has extremely low testosterone levels, even far lower than you would see in prepubescent males in most cases, someone who's genetically male, someone who never saw a whole lot of testosterone during fetal life, way below the threshold for the testosterone having any effects. So the person is born phenotypically female, female in appearance, female in external genitalia, and not internal, back to those testicular feminized males. What you find there's a vagina which goes nowhere because there's no ovaries waiting somewhere above that. There's the testes, but the external genitalia is just fine. What I think these days is the most common sort of medical ethics advice on what's done with that is the physician explains what it is, and says for all practical purposes, you have a daughter who is perfectly healthy, who's going to have a long, happy, healthy life, and simply cannot reproduce. That's the only consequence that's relevant here. And there's been a whole other world of reconstructive surgery and all sorts of very interesting, horrifying history. Okay, so back to this one. Because of the extremely low testosterone levels, because at a time of life when there's not a whole lot of testosterone around in a fetus, in a young male, and now it's below threshold for causing anything. Along comes puberty, and thanks to a whole bunch of changes in the brain, signals go out so that testosterone levels now come roaring up into the usual impossible levels in the males. And what happens here is the levels don't go up as much as they should because that enzyme is a little bit on the slow side, but they go up enough to pass threshold for beginning to have an effect. And somewhere right around puberty, this individual changes sex. This individual transmits, jumps ship, and goes from female to male as a result of these androgenic effects suddenly coming in. It's not a complete switch there, but it's somewhat of a transition. This is bizarre. Again, this is not something subtle. This is something changing completely here, and again, one single mutation. And in this case, not even a mutation wiping out the function entirely of some protein, as in PKU or testicular feminization syndrome, but now you got a protein that's just working differently, slowly enough that you get this very different picture popping out the other end. What's most interesting is you have this intersection between molecular biology and the bioendo consequences of...
Wait, isn't there a way to shoot out bits of pieces of you in order to make those things shoot discs? I thought it was C. Did they change it or get rid of this, or? If I was big enough to take over him, we would have been making big plays. We would have been making big plays. Oh, is it W? No.
This mutation, all of that, and the cultural context of it, apparently in both of these populations, people have kind of adapted to it. It's not a big deal, you know, puberty, sometimes you get acne, sometimes you get a penis, and like people just kind of deal with that. That's just part of the whole process, and cultural accommodation to interesting biology. One final example of where you can have a classical type of mutation and another version of a very mild difference. And in this case, it is not a very mild difference producing an overt disease. In this case, it's just producing something that probably differs in you from the person sitting next to you, which is you have a neurochemical system, a system of chemical messengers in your brain. If this is new stuff, hang on, it will all be explained in a couple of weeks. But this is a class of neurochemical signaling that has something to do with anxiety. A chemical messenger which decreases anxiety, and we will learn plenty about that down the line. And there's this regular class, they're known collectively as benzodiazepines. Do not panic if you've not heard that word before, and certainly do not attempt to spell it right because it's not possible to. So benzodiazepines. There are synthetic versions of benzodiazepines which people might take when they're feeling anxious, like Valium, like Librium. Valium is a synthetic benzodiazepine. So benzodiazepines are protein, and they have a particular shape, and you guessed it, there are thus benzodiazepine receptors, which could do that whole lock and key deal going on there. And what you have are differences, small single point, in this case, not mutations, but simply different versions that one letter can come in the DNA sequence specifying the benzodiazepine receptor. These are not mutations, this is just normal variability in this one particular spot can specify 2 or 3 different amino acids that function roughly the same, so that you're changing somewhat the shape of the receptor and thus changing somewhat how long the benzodiazepine stays in there and how long the signal is sent, and it's all very subtle. And what does this begin to explain? Individual differences in levels of anxiety. It's only one of a gazillion ways of explaining it, but it's part of that picture there. Here we have individual differences. This explained a very interesting finding. For decades, people often try to breed rats, different rat lines for different behavioral traits, and it makes it very useful models. There are alcoholism-prone rats. There are rats that have been bred for being smart, for being not as smart at spatial maze stuff, etc. And for years, there had been a number of rat lines that have been bred that were either high anxiety or low anxiety rats. Very useful for understanding things like, what are they useful for? I don't know, they're just cool to have around. Very useful for understanding the effects of stress or things of that. So finally, modern era of molecular biology comes in, and those high and low anxiety strains differ in the shape of the benzodiazepine receptor in many of these cases. Again, tiny little differences. Okay,
what are you doing?
So what we've got here is classical genetics at the molecular level. These tiny little changes in one single base pair at a time point deletion, insertion, a world in which it may or may not change the shape, where it could change it dramatically and produce some pretty exciting dramatic diseases where you wind up with a different gender than your chromosomes say, ones where you get more subtle differences, you change gender at your 13th birthday, or ones where we're just for the first time beginning to look at the individual differences that flow out from stuff like this, not them and their disease, but the individual variability. All of this is in the realm of single base pairs being changed. What all of this now allows us to do is translate this world of mutational changes into what does this look like evolutionarily? What does this look like in terms of explaining patterns of evolution? And you will know exactly where this goes right now. This helps explain the classical gradualist picture of little bits of change at a time. Little bits because you've got one protein which now works a little bit differently, and thus you get to ask a sociobiology question by having testosterone having a little bit more of an effect or a little bit less of an effect by way of molecular changes in this. Is that going to increase the number of copies of genes that individual has? Is it going to decrease the number it has? How's it going to fit into all of last week's science? And you will know exactly how that drill will run. Oh, if thanks to one single base pair changing, you now have a receptor which functions in a slightly different way, which makes this individual 1.5% more fertile than everybody else that they're competing against. You just wait long enough and come back, and everybody in that population is going to have that different version of it. We can run the logic there. We have variability, Darwinian variability, thanks to a mutation. We have differential fitness. We've just defined an adaptive difference somehow or other. There's a smidgen of advantage thanks to this slightly different shape. And thus we have selection changing distribution over time. The smidgen more effective version will become more common in the population. And what's also intrinsic in that is that these are slow little steps of change. This is gradualism. So gradualism is absolutely commensurate with everything we got last week, built around adaptation, competition. Every little bit makes a difference because a 1% difference in number of copies of your genes run at enough generations, and that's going to be a big difference, and all of it changing in a very gradualist, incremental way. This has popped up in a number of domains and could be really useful because it allows you, for one thing, to trace evolutionary history by looking at the changes of single base pairs, and that's allowed for looking at one really interesting gene, which we may talk about down the line, a gene called FOXP2. FOXP2 has something to do with language. FOXP2 was first identified in a family, all of whom had some sort of language communication problem, and people to this day argue whether it was mostly about coordinating the motoric aspects of speech, or was it something about grasping the symbolic message aspects of language, all of that. In any case, a mutation was found in this gene, and a whole cottage industry has emerged since then of studying FOXP2, because versions of FOXP2 occur all throughout the animal kingdom, in birds and rats and non-human primates and all sorts of things. FOXP2 popping up all over the place, and in all these different places it's got something to do with communication. It's got something to do with bird song. It's got something to do with rat ultrasonic vocalizations. Rats are constantly jabbering at each other in a range that we can't hear, and FOXP2 has something to do with all of those, and different versions of FOXP2 in all these different species. And what becomes most pertinent is when you look at those differences, you will see tiny little differences in this tree across all these different species. One base pair difference between rats and mice, one base pair difference between hawks and elephants, that sort of thing. And then you look out at humans and a whole bunch of changes. A whole bunch of changes in a very short evolutionary period. What this suggests is, no wonder this is producing some very different stuff than these guys, a very different gene. and a very different gene people now have been able to back calculate due to a series of single base pair changes somewhere in the last 250,000 years or so. And that makes a difference.
Oh,
totally cool,
shit.
unsettling experiment.
Hold on. I'm becoming the biggest guy on the fucking server. Oh, no!
That was done a few years ago, which was some people taking the human version of FOXP2 and knocking out a mouse line, knocking out their own FOXP2 and sticking in the human version and seeing what happens. And it was very interesting. They immediately sing the theme song from all the Mickey Mouse cartoons. Can they prove that? Okay, what you see is you got more complex types of ultrasonic vocalization in these mice. That's really interesting, and there's years of stuff to sort out what's going on with that. For our purposes, the main thing is here you have wound up with a very different world than chirping and buzzing and barking and all of that, and you wind up with a very different version of this gene where you can trace out how many amino acid changes it took, and every step of the way you were having some gradualist process. What's also this allows you to do is look at a footprint, an echo of what the selection was like. Okay, important concept here. So back to that business, there's 64 different ways of coding for amino acids. A couple of them are just informational, stop messages and stuff. There's about 60 different ways of coding for 20 amino acids. So on the average, each amino acid could be coded for in 3 different ways.
Okay.
So suppose you throw in a random mutation.
Yeah.
And of those, what you find is of the 60 possible mutations, 40 of them will not cause a change in an amino acid.
Okay.
Statistically, 2/3 of the time, there will not be a change.
Okay.
So in other words, if you scatter a whole bunch of mutations and you wind up seeing 2/3 are neutral in terms of their consequence, and 1/3 actually causes a change in an amino acid, that's telling you it's happening at the random expected rate of mutations popping up that are either consequential, changing an amino acid, or inconsequential, just coding for a different version of the same amino acid. Now suppose you find a gene that differs and you look at the ways in which it differs, and 99% of the base pair differences over the course of this 5 million base pair long gene, 99% of the differences make for a different amino acid than beforehand.
Whoa!
In other words, at a much higher than expected rate if this was a random process. What is this an echo of? Of very strong selection, of very strongly advantageous traits driven by these changes. Term used, this would be a mark of there having been positive selection for this trait. In other words, the changes that it has gone through over the gazillion years has not been just by random mutational rates of neutral, and remember every amino acid is coded for 3 ways. If 99% of them are consequential, there has been some major hard-ass selection going on there. Alternatively, if you find a long gene with a whole bunch of mutations and 99% of them are neutral, make no change at all, what does that tell you? This protein's function, you do not want to mess with it in the slightest. Even a minor change in the function of it, and you don't pass on copies of your genes, this would be stabilizing selection. Negative selection, strong selection, to make sure that this gene and its function downline as the protein does not change. When you look at the...
No, wait, okay, so to make sure that I understand what he's talking about, so for positive selection, I'm going to be very basic here, or very hyperbolic. Let's say that there was like a gene that coded for like the existence of the heart. Like this might be something that would fit that description, where there's like a huge chain and maybe there's a lot of mutations but they don't really impact it much. That shows that there's not a big selection for that trait because you always need the heart to survive. But for something like maybe like the speed at which you maybe say build muscle or something like that, that a slight change to this could cause the entire thing to get really fucked. That shows that that trait changes often because there's strong negative selection specifically for that trait. Is that correct? A fair summary? Wow, Crime Held, welcome to the Tier 4 Cyber Clever.
number of changes, the burst of mutations over the last 250,000 years or so that differentiated FOXP2 in humans from these other species, it's almost entirely evidences of positive selection. This did not just happen by chance. Every step of coming up with these new versions, these new amino acids in the sequence there, clearly were ones that were positively selected for. A lot of selection brought about this huge difference. Okay, so that begins to give a sense here of how these little changes can occur. At this point, we have to go through one of the all-time confusing things in genetics out there, which is something, two factoids and two sound bites that seemingly totally contradict each other.
Hold on, I'm sorry, I have to re-listen to this real quick. We'll speed it up.
Like message aspects of language, all of that. In any case, a mutation was found in this gene, and a whole cottage industry has emerged since then of studying cool unsettle of that, and you wind up with a very different version of this gene where you can trace out how many amino acid changes it took, and every step of the way you were having some gradualist process. What also this allows you to do is look at a footprint, an echo of what the selection was like. Okay, important concept here. So back to that business, there's 64 different ways of coding for amino acids. A couple of them are just informational, stop message and stuff. There's about 60 different ways of coding for 20 amino acids. So on the average, each amino acid could be coded for in 3 different ways. So suppose you throw in a random mutation, and of those, what you find is of the 60 possible mutations, 40 of them will not cause a change in an amino acid. Statistically, 2/3 of the time, there will not be a change. So in other words, if you scatter a whole bunch of mutations and you wind up seeing 2/3 are neutral in terms of their consequence, and 1/3 actually causes a change in an amino acid, that's telling you what's happening at the random expected rate of mutations popping up that are either consequential, changing an amino acid, or inconsequential, just coding for a different version of the same amino acid. Now suppose you find a gene that differs, and you look at the ways in which it differs, and 99% of the base pair differences over the course of this 5 million base pair long gene, 99% of the differences make for a different amino acid than beforehand. In other words, at a much higher than expected rate if this was a random process. What is this an echo of? Of very strong selection, of very strongly advantageous traits driven by these changes. Term used, this would be a mark of there having been positive selection for this trait. In other words, the changes that it has gone through over the gazillion years has not been just by random mutational rates of neutral, and remember every amino acid is coded for 3 ways. If 99% of them are consequential, there has been some major hard-ass selection going on there. Alternatively, if you find a long gene with a whole bunch of mutations and 99% of them are neutral, make no change at all, what does that tell you? This protein's function, you do not want to mess with it in the slightest. Even a minor change in the function of it, and you don't pass on copies of your genes, this would be stabilizing selection, negative selection, strong selection to make sure that this gene and its function downline as protein does not change. When you look at the number of changes, the burst of mutations over the last 250,000 years or so that differentiated FOXP2 in humans from these other species, it's almost entirely evidences of positive selection. This did not just happen by chance. Every step of coming up with these new versions, these new amino acids in the sequence there, clearly were ones that were positively selected for. A lot of selection brought about this huge difference. Okay, so that begins to give a sense here of how these little changes can occur. At this point, we have to go through one of the all-time confusing things in genetics.
So when he says when he says that, is he saying that on the FOXP2 thing, we would expect to look at that, and because it's a stabilized thing, we would expect that it would take a lot of mutations to like fuck that up, right? That we would call that like a stabilized thing from the selection, that it was strongly selected for, that you wouldn't expect any one random mutation to cause a change, but that rather it would be really hard to cause a change to it. Is that correct? Do I have that understanding correct? Holy shit. No? Oh, where is Nim at? I need the people that like actually like speak in complete sentences. Yes. The same guy just said no and yes. I feel like all my biomemers went to sleep. I'm going to assume I have that understood correctly.
There, which is something, two factoids and two sound bites that seemingly totally contradict with each other. You share, on the average, 50% of your DNA with a full sibling. You share 98% of your DNA with chimpanzees. What's up with that? That seems a little bit unexpected, and those are two sound bites that everybody knows. Everybody knows it goes through like basic Mendel, 50%, 100% with an identical gene, with identical twin, 50% with a full sibling. You know that song and dance by now. And meanwhile, one of the great sound bites of our evolutionary history and the mark of evolution and what in hell are you creationists thinking is the fact that, oh, humans share 98% of their DNA with chimps. This doesn't make any sense. 1 minute here to be spent on just clarifying that one that is not contradictory at all. Genes specify, by way of proteins, and now this is taking a bunch of leaps down there, traits. aspects. Genes specified, to be totally simplifying here, genes specify for antlers, for dorsal fins, for petals and pistils and stamen, for kidneys, for just totally simplifying. So right off the bat, you will have some genetic similarities between two different species in that both of them will have genes that code for dorsal fins. You're thinking about two different species of whales. So they share genes coding for dorsal fins. Whales have dorsal fins. Neither we nor chimpanzees have genes for dorsal fins. Meanwhile, we have genes coding for a pelvis that is a certain shape, which either in chimps predisposes towards being able to walk bipedal for certain lengths of time. Us as well, you don't find these genes in apple trees. They don't have pelvises that are shaped like those. So they don't have, so when you look at the human and the chimp genome, 98% of the genes code for similar kind of things. We share with chimps an absence of genes for antlers and trunks and tusks and wings or who knows what. And we share all sorts of other genes in common having to do with our shared immune systems, et cetera, et cetera. So 98% of the genes code for similar types of things. So that's where we get the 98% similarity with the chimps from. For each one of those genes, it could come in a couple of different flavors, a bunch of different flavors.
And the flavors are where you get the 50%.
Not do both you and your full sibling have a gene for opposable thumbs, do you and your sibling have the same type of gene for opposable thumbs? So suddenly that's a different world of variance on each type of gene. So when talking about the amount of DNA shared in common, the genes shared in common with another species, what you're talking about are the types of genes coding for the types of traits. When we're talking about, from last week's notion, 50%, 25%, 12.5%, one brother or 8 cousins, what you're talking about are different versions of particular genes. Important clarification. Okay, so final thing here before the break, which is once again, notice with these models, these point mutations where you can get slight differences in function, and thanks to a 1% difference in fitness and number of copies of genes, you get these gradualist changes. What's intrinsic in that, and where we get the political theme coming through, is thus there's competition everywhere. In terms of the evolution of behavior, the evolution of species, every little bit of genetic advantage will play out in some competitive way, in some little bit of an increase in reproductive success. Okay, whoa, there's a lot of apples there lit up. Well, that wasn't very subtle. Let's take a break. Whoever did that. Okay, 5-minute break. Are you slightly more fit by the rules of your population's natural selection, sexual selection, whatever, even if that gives you a tiny advantage thanks to this tiny change in this tiny gene, enough generations go by and that trait is going to become more prevalent. Final point there, intrinsic in this, is if every little bit of difference matters in terms of fitness and gene distribution, within the realm of behavior, every little bit of competition matters. A very, very intertwined sort of political, philosophical stance in gradualism as it applies to evolution. Okay, so that's been sitting there around forever. And in the 1980s, suddenly a very different model emerged. And this came from Stephen Jay Gould, who we heard about the other day. Stephen Jay Gould, and the person, the poor schnook who was always lost in this, another evolutionary biologist named Niles Eldredge, who somehow did not quite have the press that Stephen Jay Gould did, so he is lost to history, except for people who know what he's up to, and he's an amazing scientist. But Gould and Eldredge came up with a very different model. And I alluded to it the other day, and drew it right there. And their notion was that gradualism is nonsense. There are not
big changes over
gradualistic
small periods of
incremental
time.
changes. Evolution is not being driven by small gradualist changes. Instead, what their model was is that there's long periods of nothing happening, of stasis. Long periods of nothing happening. If there's changes in DNA sequences thanks to mutations, they're not consequential, or if they're consequential enough to change the fitness of one organism 1%, that's not going to make a difference. Most of the time, no change is occurring, and when it does occur, it is incredibly fast, explosive periods of change followed by a new period of stasis. Evolutionary change comes in step functions rather than smooth gradualism. Long periods of stasis followed by dramatic jumps of evolutionary change in short periods of time. Thus, they call this the notion of punctuated equilibrium. Long periods of equilibrium and stasis punctuated by periods of very rapid change. And as I mentioned the other day, Gould was a Marxist and felt that Marxist sort of stance was running through all of the ways to think about genetics. And I don't know what's up with Niles Eldredge, but that was the case with Gould. And when you look at this model, this is like classically fitting with like stasis and revolutionary change and dialectical materialism and stuff like that. The last sentence, I have no idea what I just said, but apparently that's got something to do with that stuff. And once again, we see in a very different way a political theme running through a different world's view of what evolution is about, punctuated equilibrium. Okay, so where did the idea of punctuated equilibrium come to these guys? Mainly because Gould was not a biologist. He was certainly not an evolutionary biologist. What he was a paleontologist. He studied fossils. He studied the history, the evolutionary history of fossils, and apparently, like totally separate of his large theoretical models, he was like the world's expert on the evolution of some Caribbean snail shell over the last 10 billion years or something. He's one of those paleontologists who traces lineages of evolutionary change over the course of time with fossils, fossil records as the readout. So he's a paleontologist slash geologist in some ways. And when you do that, you notice something, which is you got gaps in the record. You've got your famed missing links. You have gaps in your evolutionary record there of what the fossils look like. And you're measuring some trait or other in these snails or trilobites or whatever you're looking at. And at this time period, the trait looks like this, and at this time period, it looks like this, and this looks like a perfectly good gradualist model. And what Gould would notice is, as the field got more and more information, more and more intervening steps on a lot of these fossil histories, they would start to look more like this. And every now and then, you would see something like this in between. And from that began to suggest to him this model of punctuated equilibrium. Most of the time, as assessed by the fossil record, nothing dramatic is happening, long periods of stasis. And what allowed this to occur is that for some fossils, you have incredibly detailed evolutionary history where you can begin to fill in lines, and they wind up looking punctuated in this way. So out of him comes this whole theory that it's all about punctuated equilibrium rather than gradualism. Right off the bat, what are the consequences of that? Little genetic changes don't matter. Competition driven by the notion of little changes mattering aren't actually occurring. A model saying most of the time, all of the notions of if you figure out the right kid to kidnap when the big guy is coming at you and you'll leave more copies and figuring out exactly who to be infanticidal to, all that, it's not going to make a difference in terms of gene distribution. Evolution is not being driven by that. And out of it came this very strong indictment of the sociobiological view of what you got there is a world where it's all about competition, where it's all about hierarchy, where it's all about domination, where it's all about that. Hey, isn't that interesting that that's exactly the sort of world that these folks live in who are benefiting from this, who started this theory? Very different notion here. Competition, selective advantages, all of that, most of the time.
These two guys are teamers. That's how they got me twice. I just realized it now, though. I realized it the second time. I'm sorry. These guys are teaming. The rank 1 and rank 2 guys are fucking shitters.
Nothing's happening. So not surprisingly, all of the evolutionary types from last week did not like this one bit. And this was attacked left and right in some extremely valid ways. First form of attack is a very simple problem there, which is that you have two different disciplines happening here. You get a paleontologist and you get a evolutionary biologist, and they're functioning in completely different universes. Okay, stomach problems, and they're functioning in completely different universes there. What counts as fast for a paleontologist? These are tens of millions of years going on. Whoa, incredibly fast evolutionary change going on there. That's like 100,000 years. That's like, are you kidding me? Said the biologists, the ones who study one generation at a time. That is asinine. That is ridiculous. This is them imposing models where this has absolutely nothing to do with how evolution actually works. These geologists got completely thrown off, and these paleontologists, led by Gould, simply orders of magnitude different scale of time. Yeah, maybe in some rough approximation of what they look at, but they're not studying evolution because they're not biologists. Next critique. The next one made lots of sense also, which was you're not just an evolutionary biologist, but you're one who thinks about the evolution of the brain or the evolution of skin melanism, or the evolution of eye color, or the evolution of how many chambers in your heart you're going to have, or the evolution of any of these things that will leave no record whatsoever in the paleontological record, because all paleontology is about is shapes of stuff. Fossils. Fossils do not tell you what kind of brain was going on inside that fern. Fossils do not tell you anything about internal organs. Fossils do not tell you anything about behavior. So at this point, all of the evolutionary folks of the school from last week attack and say, yeah, what do they expect? They're studying the most boring possible things, the morphology of organisms. Ooh, just because the fact that humans over the last million years or so have not evolved sort of getting rid of the large trunk and roots that they have during springtime, and now they don't have them. Oh, that morphological change didn't occur, so obviously there's been stasis. Give me a break. What is interesting about evolution and evolutionary change, paleontologists can't pick up because all they can study are forms, morphology. So that was a big attack on these folks. So you've got the Gouldian folks, the punctuated equilibrium people, saying when you look at the fossil record, it's not gradualism. And we've got some really complete ones. And to this day, the majority of fossil pedigrees where there are very, very complete records show patterns of punctuated equilibrium. And back come the rejoinders. This is ridiculous, the time span they talk about. That makes no sense. In this period, humans evolved, doubled their brain size in the length of time that they call a very rapid evolutionary change. Their time span is completely crazy, and they can't study the evolution of anything that's interesting because they study fossils. But in lots of ways, the best rebuttal, the one that most got at these folks advocating punctuated equilibrium, would be the gradualists saying, show me a molecular mechanism. Show me some way in which you can get rapid change and then long stasis, turn that into modern molecular biology, which is occurring 2 minutes after the microevolutionary people were trashing the folks from last week, saying you need to look for the actual genes. It's not enough just to make up stories. And once these folks had assimilated what evolution looks like on the genetic level, mutational level, micro-mutational changes, they loved genetics. They loved the molecular end of it because they could now turn around to the Gouldians and say, show me the genes and show me the mutations that will account for stuff like this, because you can't account for it because we all know classical genetics and mutation. You don't get that, you get gradualism. So this was a period of enormous hostility between the two camps, and the gradualists, called the punctuated equilibrium people, evolutionary jerks. Haha. And the punctuated equilibrium people called the gradualists creeps. So Ultimately, they all got along wonderfully because they were so witty. But what you had was enormously hostile camps, and really quite hostile, because all sorts of implications spreading beyond like how fast the shape of this like seashell was going to be evolving. And when it first came on in the 80s, all of this controversy, the punctuated equilibrium people didn't have a word to say with the show me the molecular mechanisms. Show me mechanisms for mutation that will produce rapid change. And everything that has occurred since then in the world of molecular genetics that has been most striking has supported punctuated models, ways in which the micro-mutational, microevolutionary stuff of an hour ago is not what's going on an awful lot of the time. Okay, starters. So simple classical model. What we've got here is a stretch of DNA, and this box symbolizes a sequence of DNA coding for one gene. And next, right next to it, once it finishes, one of those stop codon, stop triplet signals. Right after that comes the stretch of DNA coding for the next gene and the next gene. And this is what DNA is about. It's the sequence of genes there. And what you would obviously then get is this arrow having this intervening step of that RNA stuff, but eventually producing an amino acid that produces a protein of this shape. And this is the protein coded for by this gene, this, and so on. And that's exactly how it works. That's the structure of DNA. Then, though, people began to find that that's not the structure of DNA. And what you began to get instead was something vastly more interesting, which is that for starters, when you look at coding for one single gene, it's not necessarily coded for in one continuous stretch of DNA. In other words, it's broken into little pieces. And you will have a stretch of DNA coding for the first third of the protein, and then a bunch of DNA that's got nothing to do with it. Stay tuned. Then coding for the next third, coding for the next third, that the gene was broken up into separate coding domains. And the term that was given for this was, these were called exons, and the in-between boring stuff were called introns. And this was a major finding, which made no sense whatsoever, because how are you going to get from this to then having a protein which has its normal sequence and shape, where this part was coded for by this exon, exon 1 of this gene, and this from exon 2, this from exon 3. That's a completely different sort of world of stuff. How are you going to do that? Because once you're going to make an RNA that's going to encompass all of this, and that's going to code for something completely different, because you got these introns in between there, and people then guessed something had to exist, which was soon discovered, enzymes called splicing enzymes. And what they did was exactly what you need to do to solve this, which is at the RNA level, these splicing enzymes would come along and they would snip out the part corresponding to here, and another would snip out there, and another one, as an enzyme catalyzing, would stick these two pieces together, and thus you had this utterly bizarre world in which genes, the vast majority of them, are not coded for in a continuous stretch of DNA, but instead are broken up into these separate exons. And then you need these splicing enzymes to clip out the boring in between parts, the introns, stick them all together, and you've got your functional gene. Weird. Okay, but that's how they work, though. And it was shortly after these were discovered that one of the giants in this business, a guy named David Baltimore, who got the Nobel Prize for some of the work on that reverse process of RNA viruses turning back into DNA, he was the first to really appreciate that what you got here is a potential for a lot of information. Because of, and I think he was the first person to introduce this word into thinking about it, because of the modular construction of genes. Because genes come in these separate exons. What does that begin to allow you to do? Very important stuff. Okay, so we have the same structure here, and we have a gene coded for in a modular way in 3 separate exons, And thanks to splicing enzymes, protein catalysts, clipping this out, you wind up with this. What Baltimore was the first, or one of the first, to appreciate was that you could wind up with something different there as well. You could, for example, create a very different protein, one consisting only of A and B, or one consisting only of A and C, or one consisting of B and C, or A alone, and suddenly you have this combinatorial possibility of cranking out a number of different ways of putting together 7 different ways, since you can't transcribe this gene so that there's no transcription, 7 different ways of combining these different exons, 7 different types of proteins you could generate from the same gene. This was not accepted with a whole lot of pleasure by the old guard, because intrinsic in the know the DNA sequence
These
specifies
teamers, dude.
amino.
These teamers are so fucking stupid. Fuck me. It sucks because you can't see. I can't see all around what's going on with the other dude. He broke himself so that his friend could eat him, so that he could eat me.
Acid, protein shape, protein function. Intrinsic in that is one gene specifies one protein. One gene only specifies a single protein. One protein is only specified, coded for by one gene. And suddenly this modular business allows you, with one gene, to generate 7 different kinds of proteins. For starters, how could that possibly work that way? Just on a nuts and bolts level, all you need are splicing enzymes that work a little bit differently in different parts of the body. One that will splice this off and is attached to an enzyme that will degrade this, while the splicing enzyme here does, and what have you just produced? You'll get this one, coupling of splicing enzymes with degradative enzymes, and suddenly you've got a means to have tissue-specific expression of genes. The same gene will produce different types of proteins in different parts of the body because of splicing enzymes working differently. Then, just to confuse things even more, people began to note that there would be some splicing enzymes and genes where they would splice at a different point. And you would now have a gene with A and A prime and other splicing enzymes that would cut at other points, one single sequence of DNA generating all sorts of different types of proteins in different parts of the body, at different times of life, under different circumstances, in different individuals in different ways. Suddenly, there's a lot more information floating around in there. So that was a huge, huge breakthrough in the field, understanding this modular basis of gene construction. And for our purposes right now, what the most interesting consequence of that is it's no longer one gene, one protein. One gene instead can generate all sorts of different types of proteins, different settings, different circumstances. The next thing that was intrinsic in this model.
What he's saying here is that basically like one gene might not contain all of the information for a protein, but when you plug that gene in with another gene, or when that gene is in a different area, that in combination with something else, it'll code for proteins, right? Or code for amino acids that'll create proteins is essentially what he's saying, right? So that like a singular gene can be plugged into a lot of different other genes to get different types of outcomes. Oh, I need to watch these earlier before my biomemos go to sleep. Fuck. That's essentially what he's saying, right?
Model that's now been trashed of one continuous gene. What we just figured out is instead you can have the intron, exons, all of that. The next thing that went down the tubes was looking at, well, how much of DNA is actually devoted to coding for amino acids? And the answer was obvious, like 99.99%. Each one of these would just have to have a stop codon, a stop signal at the end, and otherwise this was just a continuous flow of information once you have factored in these intron things. Okay, so they're part of this gene, but immediately starts the next one. The next major discovery was one gene would very rarely start immediately after the next one. There would be long stretches of DNA in between that didn't code for a protein, non-coding DNA. That's mighty puzzling. Like, what's that, just junk or stuff? And around that time, the phrase junk DNA was actually floating around, people trying to make sense of this. And when people sat and started actually like doing the numbers, out came a number that knocked people on their rears. It was so flabbergasting. 95% of DNA is non-coding. 95% does not code for a gene specifying a protein. In other words, in between here, on the average, would be a stretch of DNA 19 times the length of that, or whatever the math winds up being, and suddenly calling that stuff junk DNA started seeming a little bit tenuous, because 95% of your DNA just can't be packing material for the whole thing. It's got to be doing something. And during that period came sort of the insight into this, that all that intervening non-coding stuff was, what was that? That was the instruction booklet. That was the instruction booklet on when to activate these genes. That was the on and off switches for turning genes on or off. Upstream in the non-coding domain, just above a particular gene sequence, would be the information for when that gene is activated. Activated when it makes RNA into protein, all of that. And upstream of that are the on and off switches. Implication right there off the bat, which is Crick was wrong. DNA sequences are not the starting point of the central dogma of life, and DNA is the rule giver and all of that. DNA is being regulated, genes are being regulated in some other way, and where 95% of DNA is being devoted to regulation of the genes. DNA has no idea what it's doing. DNA is a readout that's under the control of all sorts of other factors. And out of this emerged the really, really important concepts of regulatory sequences upstream from genes. So here we have a long string of DNA coding for this gene, which happens to come in 2 exons, and it is like the space between a galaxy, how long the non-coding is going to go on until the next gene is back there. So what's going on in this stretch just upstream for this gene? Things that were soon being called stuff like promoter sequences or repressor sequences, things, stretches of DNA that coded for switches rather than coding for protein, that coded for things coming in to the neighborhood of the DNA and binding to some of these promoter or repressor sequences and then turning on, or in some cases off, the transcription of that gene, the process of the gene generating proteins. You would have promoters sitting there. And this is overly literal. It would be just a sequence of DNA, which thanks to that sequence would have a certain subtle micro shape, and along would come something which happened to be able to fit perfectly into that spot. And if and only if this molecule, usually a protein, bound to this promoter, suddenly that would trigger a whole bunch of enzymes to come in, which would start the process of transcribing this gene. This would be the switch, and this is the thing that just turned the switch on. And these things that turn the switches on, or often in some cases, were soon called transcription factors. Totally critical concept in there. The fact that vast stretches of DNA don't code for anything. Instead, they have the instruction booklets. Here's how you turn this gene on or off. Send in this molecular messenger. Send in this type of transcription factor. And if it shows up, binds to here, you now activate the transcription of this gene, the process of turning that gene, making proteins derived from it. This is where the information was. And this is not DNA knowing what it's doing. This is outside regulators coming in. So immediately, that has made life a whole lot more complicated. Next complications. You could have different genes scattered all over the place that would have the same promoter upstream of it. What would that mean? In comes a transcription factor, and it doesn't activate the transcription of one gene producing one protein. It activates the transcription of a whole bunch of them. In other words, now suddenly we have messengers that can trigger activation of genetic networks, entire arrays of proteins being produced, all with a functional similarity, driven by the fact that all of them have the same promoter upstream. So suddenly you have the possibility of the same promoter being upstream regulating more than one gene. That is the general rule. Flip side of it. Any given gene, for example, could have a bunch of different promoters responding to different types of signals. So now, suddenly, you have this gene which can be transcribed under this circumstance or under this circumstance, and both are ways of turning on activation of that gene. And in this circumstance, the same promoter is found in genes A, B, and C, somewhere at the other end of the chromosome. And in this case, this promoter is found on genes D, E, and F down there, different networks. So the same sort of transcription factor logic, once you can have different promoters upstream of a gene, and once you can have the same promoter upstream from multiple genes, you suddenly have the ability with different transcription factors to activate entire networks of gene expression. Okay, so what this has allowed you to do is completely trash this notion of DNA knows what it's doing. DNA is just the readout, and who knows what's going on? Whatever is controlling the transcription factors, whatever is causing them to do their thing. And this could be a world of influences. And to be absolutely accurate, this requires introducing the word environment. This is environment having something to do with genetic effects. This is going to be a way in which environment is interacting with genetic elements, environment determining which of these are doing what. What would that look like? Sometimes environment could be the environment in the rest of the cell. So you got your DNA there, and your stretches and all of that, and the promoter, and we could do that by now. And something is occurring inside the cell, which activates some transcription factor, and then you go change a genetic event going on in there. The cell is running out of energy. There's various ways to construct sensors. In other words, evolution has come up with a bunch of different ways in which cells can respond to signals of low energy, and that will activate some transcription factor, which will go and bind to a bunch of promoters, which produce proteins involved in taking up more energy from outside the cell, transporting in more, using energy more efficiently. So what we have here is an environmental regulation of genetic effects, environment, the rest of the environment of the cell. The DNA doesn't know what it's doing. The gene doesn't know what it's doing. Events going on elsewhere in the cell is what's regulating what's up. Some of the time, the environment can be even more far-flung. And in this case, it's now events going on outside.
This is so laggy.
In this case now, you've got the cell with its DNA, and now instead you have a chemical messenger coming from somewhere else and binds to its receptor like a lock and key, and as a result of this binding, something happens here, which does something here, which does something here, which eventually activates some transcription factor, which goes and does its thing. Now we have gene expression in the cell being regulated by the environment somewhere else in the body. Somewhere else there. What would be a classic example of that? This is what a whole lot of hormones do. Hormones go floating around and they affect cells everywhere throughout the body. By definition, a hormone is a blood-borne chemical messenger. So you could secrete some hormone out of the top of your ear and it will affect things in your little toe, very far flung. Hormones will bind to their receptors, lock and key. Most hormones are protein in nature and trigger what's called a second messenger cascade jargon. Don't worry about it, just get it conceptually. Activate some transcription factor, deactivate some other transcription, and suddenly events going on, 14 counties over there are regulating what proteins are being made in this cell. What would be an example of that? Testosterone, for example. Testosterone secreted from the testes, traveling far and wide and eventually binding to androgen receptors on muscle. And what happens there is, through a pathway like this, turning on the activation of genes, coding for proteins, all sorts of structural proteins that will make that muscle cell bigger. Your muscles are getting bigger thanks to testosterone. Look at this. Events occurring a gazillion cells away in the testes, a messenger here determining what gene expression, what gene activation is happening. Sometimes, though, the environment could be completely outside the organism. Like that. Sometimes you can have, for example, a messenger from the outside world. What sort of messenger? A scary sight, a bit of sensory information, whatever. Olfactory. Olfactory, suppose some odorant comes in, a pheromone. You're a female rat who has recently given birth, and the pheromones from your babies come floating in and bind to receptors here. Very similar principle again. And that causes them to activate something, and some cell down there. And eventually, there's a cell here that controls some muscles, and it makes your eyes dilate because you love the smell of your baby, and you're just, I don't know if rats have their eyes dilate, but humans will in many of those circumstances. Aha, how do you do that? You just change some structural proteins that did this or that, or suddenly this female is making some hormone like oxytocin as a result of smelling her baby. What have we got here? We got something going on in the outside world regulating what's going on with genes. Genes as the central dogma of life, as the information giver, nonsense. Events going on in the rest of the cell, the rest of the organism, the rest of the universe are determining when genes activate. So what we see now is a much more interesting world of regulation of gene expression. First, the modular ability for one gene to generate all sorts of different proteins, which brings up an issue after a while is, does that count as one gene? And people argue over that. And because of this 95% regulatory sequence business, the most interesting stuff going on with DNA is not what the protein is like, not what the protein does, not how well the protein does it, but when it does it, in what contexts. And what we've just introduced are if-then clauses into this whole world. If you get a signal that there is low glucose in the cell, then you begin to make proteins related to glucose uptake. If you have the smell of your child come in, then you activate this path. It's not changing what the protein is like, it's changing context. And I think what we will see is context is vastly more interesting than whether this protein is a little bit more like this, a little bit more like that. If it is being expressed instead at a different time, in a different place, in a different context, that's much more interesting. So what does that set us up for here? Now beginning to see the organization of this. By the way, 95% of this whole stuff here, I don't know what percentage is accounted for by identified promoters and repressors and stuff, but it's a tiny percentage. What that means is there's regulatory stuff going on that no one has a clue about. The main thing though here is modulatory structure to genes, introns, exons, and this whole world of the environment regulating when genes are turned on and off, a whole world where you can generate completely different proteins, not a protein that's a little bit more this way, a little bit more that way, in completely different contexts. So all we need to do now is begin to stick this into the molecular biology of mutations and evolutionary change. Where does this begin? Let me just see. Oh, no, before we do that, that's not what we're going to do. We're going to look at one more level of regulation here. Okay, so you got your DNA, and we already know this whole business, what's telling it what to do. You can have transcription factors coming in, all of that, these interesting implications. DNA, let's see, for our purposes, is protected in sort of layers of protein that are just sort of structurally stabilizing. This is not really what they look like or quite what they're made of, but they're called chromatin. There's just stuff that stabilizes the DNA because these are wispy little things. And one of the things that, of course, they need to do is they're enwrapped around the DNA stabilizing. You got some transcription factor coming in from somewhere else in the cell. It's got to be able to get down to the DNA. And thus you have a whole world of chromatin opening up to allow transcription factors to get through. And thus you have a whole world of what's telling the chromatin to open up where and when. Suddenly a whole world of regulation of whether the transcription factors even have access to the DNA. So you can have tons of a transcription factor and all set to transcribe something off of this. And thanks to conformational changes, folding or unfolding of chromatin, you're regulating whether the transcription factor can even get through. And thus there's a whole world of stuff that changes, chromatin modeling and remodeling. Additional step here, one that's really interesting, is you can do things, circumstances arise, the environment can do things, where you change the structure of chromatin around a particular gene in a way that makes it easier to transcribe or harder to transcribe, and you can essentially make that change permanent. You could permanently do something in some particular stretch of chromatin so it will never open up again to allow the transcription factor in. And what you have just done, jargon, is you have silenced that gene. You've silenced it permanently. And people know a lot of the mechanisms for how this occurs. For those who care about such things, the process is called methylation. This is a little bit different. That's occurring with the DNA itself. But this is silencing of genes by structural access of transcription factors to it. When does that occur? There's all sorts of circumstances early in life where you will change the permanent accessibility of some gene and transcription factors. You will cause long-term, lifelong changes. As but one example, and one that we will look at a number of times down the pike there, in rats, the mothering style of the mother rat will cause chromatin changes, permanent ones, in some of the genes related to stress hormones. So that certain types of mothering, how often you lick the baby and other rat mother type stuff, will regulate how readily some genes will be turned on for the rest of your life. This is early experience. This is molecular mechanisms for events early in life lasting forever. A lot of these turn out to be a little bit reversible, but for our purposes, lasting forever, this is a whole new field called epigenetics. Genetics is all about DNA sequences. Epigenetics is all about regulation of...
Oh, shit. When he says that you're not able to get in for transcription purposes, does that literally mean, like, Oh, I don't know how babies, I guess, fit into all of this. Could you silence a gene in such a way that it's not able to be replicated in, like, sperm and, like, ovum and all that shit or whatever to create, like, babies, or no? I don't know if that applies to this or not. Where's Nim at in chat?
Access to DNA sequences, things of that sort. So suddenly this epigenetic world is entirely capable of overriding anything going on at the transcription factor end. Just to give you a sense of this, researcher, this is a guy at National Institutes of Health, a guy named Steve Sumi, who studies primate social behavior, and what he has shown is in monkeys, in one part of their brain, you change the style of mothering that monkey is subject to as a baby, and you will change the conformational access state.
Chromatin is what is denying entry. Yeah, what exactly is the chromatin denying entry to? It's denying entry into sections of the DNA, right? But that has nothing to do with the baby meme or anything. I shouldn't speak anything about epigenetics. I only know what I've read on Reddit about it. I guess we should wait until we actually get to it. I'm guessing in one of these lectures, so never mind. Yeah, that's what I've heard. I've read memes about, like, epigenetics can actually be passed on generationally, but I don't know the process of which any of that works, so I should probably just wait till we get to it in the lecture rather than go by assumed to be true information I've read on Reddit or some shit.
Of 4,000 different genes. Enormously influential there. Enormous arrays of ways of regulating where it's not genetics, it's epigenetics. And this has given rise to a great phrase: fertilization is all about genetics. Development is all about epigenetics. And what epigenetics is about are ways in which the environment not only can regulate what's going on with this gene right now, but can cause lifelong differences in the ability to access genes. So this is an enormous array of levels of regulation. Splicing enzymes determining how your exons get mixed and matched, generating all sorts of different proteins. Transcription factors representing the things that turn the switches on and off, and the array of switches is far more interesting and plentiful than the gene itself. Transcription factors reflecting what's going on in the outside world, like in the rest of the cell to the other side of the planet. And finally, this whole additional level of regulation where some of the regulatory consequences here can be lifelong. Enormous array of levels of regulation. And an enormous number of ways in which just the DNA sequence of the gene itself is not very interesting. That determines the shape of the protein. But all this other stuff is, where is it expressed, when is it expressed, in what contexts, what sort of if-then contingencies, whether it is ever expressed after a certain childhood event. All of that much, much more interesting. So what we need to do now is transition to thinking about evolution, mutations, on the level of all the stuff we just heard about. And all I will leave you with, and picking up on Wednesday, is think about, what if you get a mutation in a splicing enzyme? What if you get a mutation in a transcription factor? What if you get a mutation not in the letters coding for a gene, but coding for a promoter? What happens in all those cases? And suddenly you begin to see a world in which you can get stuff that's not purely gradual.
For more,
that's not purely what? Oh, that's not purely gradual. Oh, so because we're making the case for the punctuated equilibrium model of evolution, right? That if you get an error in some of those transcriptors or whatever, that you could get some crazy ass shit. Okay. Tomorrow is Molecular Genetics 2, Part 5. Wait, what the fuck? Do we have a guest lecture? Are these TAs? Who is this cuck?
Oh, you would respond glutamate, and then you would be right.
How old are these lectures? What the fuck is this hairstyle? This is from 2010? What? Whoa.
And so you can imagine that for...
Who is this guy?
As opposed to a kind of...
This doesn't seem that relevant to human behavioral biology. How is it not relevant, my dude? I think right now what we're going over is the basic ways of just the basic ideas of genes and how they express themselves, and the idea that there is a multidisciplinary approach to take insofar as deciding how a gene expresses itself as a phenotype, right? So right now we're looking at like the individual memes, like the genes, and all that shit and all the things that impacts the ways in which they express themselves, right? That it's not just the DNA is in control of everything, but that the environment of a cell or the environment of a person or the environment outside of a person can all have impacts on ways that a gene expresses itself, that other things are in control besides necessarily the DNA, which all plays into human behavioral biology, right? I think it's all connected. Okay. The behavioral genetics ones are about twin studies? Oh shit, really? That's still like 3 lectures away though. Whew. It's just really detailed for a starter course is what I meant, but I suppose it will all fit together eventually. Yeah, after hearing these terms for so long, I mean, eventually they'll stick more, right? And then if you ever explore deeper into these, I'm sure that you'll remember hearing about them, so... I don't think it's necessary. She's still awake? What the fuck? I don't think it's necessary to remember every single little meme. I think that taking away the broad concepts is probably the most important thing. Well, I don't know what his tests are like. I would be really... Do you think that there are like midterms or finals that are ever released, like sample ones for this course? I'm curious what the questions are based on these lectures, on how specific he gets. I guess like the main thing to take away from here is that a single gene can code for many different proteins depending upon where it's located or how it's expressed. That a lot of our DNA, I think he said 95% of our DNA is just instructions for how to read it or signaling for how to interpret it or whatever. That it isn't all pure. It isn't all pure like genes and shit that are coding for proteins, but rather things that are instructions for like when to turn shit on and off or whatever. Something like that, fuck. And that, yeah, what else? That amino acids are made, that things come in and read the DNA, that, we got into the silencing, methylation, I think is what he mentioned. Methylation, the idea of permanently silencing a gene, or at least almost permanently silencing it, that you can permanently impact a gene's ability to turn on or off to code for certain, amino acids for the rest of your life. He called that epigenetics. I don't know if that... Any plans to marathon near? Yeah, hopefully tomorrow we can start. Wow, hollow wishes.
My dude, what are you doing down this rabbit hole?
We're just reading or just learning about memes, I guess. What the fuck? The fuck is this shit? Wait, are these people that submit their notes for the class? That's so cute. Do you have to like pay to like to see other people's notes? Oh yeah, okay. Did you order your new streaming PC?
Many serious game streamers use a second PC actually. So
what?
got one for gaming, and then another one dedicated to encoding and interacting with their audience.
Interacting with their audience. All right, I'm watching this and I'm going to sleep. It's way past my bedtime.
So we were brainstorming and talking about some cool projects that we've done in the past with Unraid and virtualization technology, and, well, let's just say the old light bulb went off.
Wow.
What if we could build, using an Intel high core count Core i9 processor, a 2-in-1 solution, splitting a single tower with a single motherboard effectively in half to perform both of those duties? Virtually, of course. Would we end up with a viable day-to-day streaming solution? Well, thanks to Intel's sponsorship of this investigation, we are about to find out. Okay, so let's start with this. Why would anyone want to run a virtualized 2-in-1 setup as opposed to just having 2 separate machines? I mean, bare metal's more efficient, right? Let's take a look at a few reasons why it might actually be a cool idea. First, and most importantly for many, is cost. Unless you already have a second computer to dedicate to encoding, you would still have to buy a GPU, RAM, and storage for 2, but you could save the considerable expense of many other components. In addition, your total power consumption and heat output will be a touch lower than running separate PCs, meaning your energy bill will be lower too. The second reason to go virtual is space. While it's not a concern for some, if you're streaming from a college dorm or a small apartment, this means one less tower taking up space in your gaming area. Not every virtualized 2-in-1 has to be this big. Reason number 3, versatility. Since the gaming and streaming setups are virtualized, not only are any potential software issues confined to each VM, but they can also be much more quickly and easily backed up or cloned once you're set up, so in the future you can easily hit the reset button in case anything should go wrong. Also, thanks to the magic of Unraid, your gaming/streaming machine can act as a file and media server for the rest of your household. So, the plan then is to use a portion of our CPU to encode our video stream using x264 with high quality settings. That way we can avoid the potential for scheduler conflicts without manually assigning CPU affinities in Task Manager like we would if we were doing both tasks on one OS. Well, okay, Linus, this all sounds great, but there's gotta be disadvantages, right? Well, there is the fact that when using virtualization, some devices can't be easily allocated to a VM. Our onboard audio, for example, shares an IOMMU group with the chipset and essentially cannot be used. Same story with USB ports. Each grouping of ports has to be on a dedicated controller in order to pass it through so that you can hot plug your devices, something that we pretty much take for granted these days. And finally, while it's possible to run Unraid without its own video card, it's a royal pain. So we grabbed a dedicated sound card for our gaming VM, an extra USB controller, we're using one of the
this already sounds way too complicated. Fuck.
onboard ones for the other VM, and an El Cheapo third graphics card for Unraid. So we are ready to
I'm sorry, I'm retarded. Why do we have to create a RAID array for virtualized setups? I missed it. I don't want to go back. Can somebody explain why we need a RAID array for the virtual setups?
testing, we found that gaming performance was actually best when we assigned 4 cores and 8 threads to our gaming VM. And in some configurations, we actually found tests that performed better in a virtualized environment compared to an equivalent configuration running on bare metal. What? So this is the kind of thing that we encounter sometimes when we're running on the bleeding edge. We spoke with Unraid's developers about the issue, and they're putting together some information to share with the Red Hat and KVM hypervisor folks so that hopefully we'll see a resolution by the time games demand more cores for optimal performance. But for the time being, our classic 4-core, 8-thread setup manages a negligible performance hit compared to bare metal, and our streaming VM can suck up those extra cores anyway, so that it can be used for heavier video editing and faster encoding for those edited videos that can later be uploaded to YouTube or other video on demand sites. Alright then, so let's show off the rest of the setup. We're using the utterly unique Level 1 Text DisplayPort 1.2 kVM from Wendell and his team. This gives us support for high refresh rate monitors like ASUS's 240Hz ROG Swift PG258Q and easy switching between our VMs, saving us from using 2 keyboards and mice. Though it should be noted that having an extra one on hand is pretty useful in some cases. We're using Corsair.
Wait, is dude, I don't know anything about VM. Unraid virtualizes CPUs. Oh, okay. This is some VM shit that's way more complicated than I mean, I've never virtualized shit before. I guess I shouldn't say.
There's massive Obsidian 900D tower thanks to its ample cooling capacity, and we're powering all 3 of our GPUs, the 1080 Ti for gaming, the 1050 Ti for streaming, and that basic one for Unraid off of an AX1200i power supply, and we've thrown in a monstrous Unraid array of 7 12TB Seagate Ironwolf Pro drives for 60 terabytes of protected storage that's visible to both VMs, not to mention, as I said before, anyone else on the network who needs it, allowing us to record our streams basically indefinitely so we can make them epic frag vids after we're done streaming.
Epic frag
Finally,
vids.
For our capture card, another surprise, no capture card. So we actually originally had planned to use one, that 4K one from Elgato, but this ended up being way cooler. So our gaming VM is actually transmitting its video and audio feed to the encoder VM using the low-impact OBS NDI plugin, which runs with very little quality loss over our virtualized 10 gigabit network connection between our VMs. Then from there, we've set the streaming machine's output settings to Twitch's ultimate 1080p 60 FPS quality setting. And would you look at that? After a few command line tweaks, we could not only encode on the fly, but do so at x264's slow preset, which is 6 notches up from the default potato quality ultra fast. And to validate our original hypothesis, is there some benefit from separating them in VMs, we pushed our 11 hyper-threaded cores to their limit in the streaming VM to the point where we forced our encoded video to start dropping frames. Then we went back to the gaming VM to see if those resources were effectively being isolated, and it was still running games like nobody's business. So what have we learned then on this journey?
Oh,
well, for
that's
one,
really
a
sick.
virtualized dual-head system, with all of its shortcomings, has some advantages. Operating our 2 virtual machines was about as transparent as running 2 completely independent systems.
Do you think I could do that shit? What chip are they using? Are you using the 7980XE, do you think? Or do you think they're using a smaller i9? Click. They're using a 7960X. They're not even using the big ass, dude.
Whether
79
via
mm.
KVM switch or with 2 full sets of peripherals. This is a use case for many core systems that we've been excited about for years now, and one that has many applications beyond game streaming that is just going to get better in time. And second is that with superior codecs such as X265 on the horizon that are better optimized not only for image quality, but for better thread and memory utilization that happens to mesh great with Core i9's new cache design, you'll be able to push the quality even higher with a setup like this. All we need to do then is wait for Twitch and mainstream broadcasting software to support it. In the meantime, though, we've still built one of the coolest X264 streaming setups out there, so thanks to everyone for their help with.
Look, it's dropped in price, guys. Only 13 left. I'm getting this soon. Can you explain why that's really sick? No, I don't feel like it. Okay, basically, if you run games and stream on the same computer, even if you've got the affinity of the cores set to your different tasks, it seems like there are still some sort of sub-architectural resources or something, if that word even makes sense, that's being utilized by the streaming thing that makes your games run like shit. So basically what I'm saying is that no matter how strong your CPU is or how many millions of cores you have, it seems like if you stream and play games on the same PC, it always causes a little bit of like frame lag or something, some annoying shit that makes me not want to do it. That's why I switched to a 2-PC setup. Even if your CPU is only like 50%, streaming and gaming on the same PC just always felt bad. It felt like there was a lag or something that I couldn't explain. But apparently in their virtualized setup, they didn't run into that at all, which is cool. That's why it's cool. Aren't you already using 2 systems? Why not upgrade to 10 gigabit and use the plugin they use to push low latency? What the fuck are you talking about? 10 gigabit what? How is this better than a 2-PC setup? Oh, I never said it was better. I just said it was really cool. Okay. I'm gonna host our buddy. Why is she still streaming so late? Is there like some special meme going on? I'm Lily Pichu from YouTube. Do you think on YouTube it says I'm Lily Pichu from Twitch? I bet money they did run into it. They're just retarded? Oh, maybe. I don't know. This is retarded. Imagine having to troubleshoot that shit. That's how I felt like when I was first streaming. Remember that? There's no help on troubleshooting anything. Nobody cared about anything you ever did. You were all on your own. Okay. I'm Lily Pichu. Alright, I'm hosted. Our buddy. Stay safe, Ripperino, Cappuccino, Pappuccino, Hoppuccino, Moppuccino, my dudes. Signing out. I'll see you guys later. Good night.